Hi, and welcome to my flash talk on investigating the impact of environment on antimicrobial resistance. Antimicrobial resistance is a growing global issue and has already killed 700,000 people annually. Therefore, research in this area is of vital importance. My research focuses on testing antimicrobial resistance in more clinically realistic models. We wanted to test the antimicrobial resistance of Pseudomonas aeruginosa when grown in two different environments, which were a synthetic cystic fibrosis media seen on the left and an ex vivo pig lung model on the right, which allows for the formation of biofilms. We grew two strains of Pseudomonas aeruginosa, PA14 and LesB58 in both environments. We performed a standard antimicrobial test in the cystic fibrosis medium, and with the lung, we dissected the bronchi, infected and incubated them for 2 days to allow for a mature biofilm formation, and the lung pieces were then transferred into antibiotic. The concentration of colistin needed to kill the two Pseudomonas aeruginosa strains in the lung seen on the right are significantly higher than when tested in the cystic fibrosis media. This difference in resistance is possibly due to the biofilm formation on the lung piece. In order to test whether biofilm formation was key to the higher resistance in the lung, I tested two biofilm mutants, Pele and Gake, with the PA14 wild type. The biofilm mutants had similar MIC values when performed in cystic fibrosis media. In the lung model, the biofilm mutants survived at the same levels of antibiotic as the wild type, suggesting that the lung environment itself increases resistance in addition to biofilm formation. In conclusion, the lung environment leads to higher resistant rates in Pseudomonas aeruginosa strains, and that this higher resistance is not solely due to biofilm formation, but is aided by the lung environment itself. Thank you for listening to my flash talk.
Hello, I'm Jacob, a third-year PhD candidate at the John Innes Centre in Norwich, and I also sit on SFAM's Policy Subcommittee. Today I'm here to talk to you about my project, where I've extracted potentially novel antibiotics from bacteria isolated from the nests of fungus-growing ants. Ants are one of several organisms that have discovered agriculture. This image below is of the leafcutter agriculture system. The leafcutter ants chop up plant material from their home in the Amazon rainforest and bring it back to their nest, where they use it as a substrate to grow their food fungus, Leucoa aerugarius. Because this Leucoa aerugarius is in an environment which the ants have specifically constructed using ventilation systems and water irrigation systems to optimize the growth rate of the fungus, it is susceptible to invasion from the pathogenic fungus Escovopsis, which is a specialized pathogen only found in the ant colonies. To protect their ant colonies from this pathogen, the ants have developed a symbiotic relationship with actinomycetes, and most notably Pseudonocardia, which produce antibiotics for the ants, and in exchange the ants provide the bacteria with a carbon source. I have samples and high-quality genome sequences of 18 actinomycete strains from a variety of ant colonies. I have screened all of these strains on 31 different growth media against Bacillus subtilis, E. coli, and Candida albicans for a total of 6,000 petri dishes to determine the actinomycete's ability to inhibit human pathogens. Bioassays were performed to determine the activity of the strains, as you can see in the image on the left, and this activity was compared to the anti-SMASH data. Anti-SMASH was used to analyse the biosynthetic gene clusters present in the genome sequences of the actinomycetes and could be compared to their activity profiles to see if there's any known antibiotics causing the activity seen. Several strains show unexplained antimicrobial activity, and these strains were grown on plates and had their antimicrobial agents extracted. Several of these antimicrobials have been successfully extracted and have been able to demonstrate activity after being dropped directly onto a plate inoculated by the pathogen. Work is underway to determine the chemical structure of these compounds using LCMS, HPLC, UV-Vis and similar analytical techniques. I'm also hoping to determine the mechanism of action of these compounds, some of which I have demonstrated the ability to inhibit the growth of drug-resistant strains of E. coli and MRSA. With that, I'd like to say thank you for coming to my talk. Please feel free to tweet me with any questions and enjoy the rest of the programme.
The misuse of antibiotics directly contributes to the rise in antibiotic resistance. Currently, many antibiotics are prescribed blindly, and this is because many tests to detect resistance are slow or laborious and require access to well-equipped labs. We've developed a miniaturized dipstick version of the gold standard broth microdilution method. Each test strip contains 10 parallel capillaries containing dilutions of lyophilized antibiotic. Each capillary holds 1 microliter sample volumes, 100 times smaller than conventional BMD. The bacterial sample is mixed with the metabolic indicator rizazorin, and the dipsticks draw the sample up by capillary action. This splits a single sample into 9 dilutions of antibiotic and a growth control. The test can be analysed by following the colour change from blue to pink. Blue indicates inhibition of growth and susceptibility, and a colour change to pink indicates bacterial growth and resistance to the antibiotic present. The colour change is easily visible by eye and can be recorded using a smartphone camera. Individual dipsticks can be arranged into a high-throughput comb compatible with a 96-well plate. This allows 12 microcapillary BMD tests to be run simultaneously and increases the number of isolates tested for a single multi-well plate from around 6 to 96 isolates. We have validated our microcapillary BMD test against 22 E. coli isolates, using 6 different antibiotics commonly prescribed for urinary tract infections. We found the dipstick gave 100% essential agreement in MIC and 96% categorical agreement compared to the gold standard broth microdilution method. We then combine the microfluidic dipsticks with time-lapse imaging using a simple in-house system. Using this system, kinetic data from over 2,000 data points can be captured, and allows us to look at earlier time points and automate analysis. When determining the MIC after 6 hours, we still achieved 91% categorical agreement compared to the gold standard method. Fast, high-throughput automated diagnostic devices to determine antibiotic resistance are crucial to allow rapid clinical decision-making, provide effective treatment and reduce the inappropriate use of antibiotics.
Hello, I'm Brendan Gilbride, and my presentation is on catalytic ferromagnetic gold nanoparticle immunoassay, which is used for the detection and differentiation of Mycobacterium tuberculosis, MTB, and in bovis, Mycobacterium bovis. TB is a respiratory disease which is spread by airborne transmission, which includes spitting, coughing, and sneezing, and it has symptoms which include fever, night sweats, and weight loss. Just over a quarter of the world's global population, 2.2 billion people, are infected with TB, and it is the leading cause of death worldwide from a single infectious agent. However, even these figures are likely to be an underestimate because of inadequate disease surveillance in poor regions. MTB is the main causative organism of TB in humans, but in bovis, which causes bovine TB, accounts for an estimated 10% of all human TB cases, zoonotic TB. The TB disease caused by MTB and M. bovis is indistinguishable clinically and pathologically. Both organisms share 99.95% genomic similarity, have identical 16S ribosomal RNA sequences, and can only be speciated using expensive molecular techniques such as real-time PCR. Consequently, speciation is rarely undertaken, and therefore human TB infections are routinely considered to be caused by MTB. Speciation is, however, essential because M. bovis is intrinsically resistant to pyrazinamide, a first-line anti-TB drug, and the misdiagnosis of zoonotic TB as human TB can result in treatment failure and contribute to the development of antibiotic resistance. And at present, no TB immunoassay can differentiate between MTB and M. bovis. In this research, two immunoassays were developed, one for MTB and one for M. bovis. Novel antibodies were used to biofunctionalize the ferromagnetic gold nanoparticles by direct coupling, which enabled the formation of bioconjugates which demonstrated greater peroxidase-like catalytic efficiencies and physicochemical stabilities than conventional HRP enzyme. The bioconjugates were then resultantly incorporated into a sandwich ELISA type immunoassay, which enabled simultaneous immunological sensing and differentiation of MTB and M. bovis at concentrations of 1000 CFU per ml. The addition of TMB substrate to samples containing captured bioconjugate target cell complexes resulted in a blue-coloured oxidation product of peroxidized TMB. However, if the sample remained colourless, TMB remained unoxidized because no target cell was present or bound in the complex. The absorbance intensity was proportional to the amount of bound detector antibody complex. The greater the signal, the greater the amount of bound cell complex. The novel assay represents a potentially useful tool to use to reduce the emergence of multi-drug resistant TB. Thanks for listening.
Hello, my name is Jonathan and I'm going to talk to you about the development of methods for the environmental surveillance of Salmonella typhi. Initially hosted at Public Health England, several media were identified through literature review. 16 of these were eliminated due to logistics and shelf life, leaving 8 agar and 5 broths. Combinations of these media, which we called pathways, were created. Pure cultures of 18 clinical strains of S. typhi were provided by PHE's gastrointestinal bacteria reference unit and were used for growth comparisons. Mixed culture challenges followed using NCTC strains of common environmental organisms provided by PHE's Food, Water and Environmental laboratories. After these tests, 8 pathways remained, all of which included a modified version of chromogenic agar called case from Neogen. The unmodified agar inhibited S. typhi growth, but working with the manufacturer, the causative agent was identified and removed. These 8 pathways were narrowed down to one final pathway when trialled in Blantyre, Malawi, and was selected for its efficacy in isolating salmonella of all kinds from local river water prior to our pilot field study. A real-time PCR assay was adapted into a multiplex assay from PHE GBRU's hazard group Salmonella PCR, which were designed for the detection of S. typhi and S. paratyphi A, B, and C. From this assay, 4 primer and probe sets were selected. TTR, a pan-salmonella target found in both species of Salmonella. STAG, this target is commonly used for S. typhi detection but has been shown to be less specific when tested by in silico PCR. TVIB, this target is less sensitive than STAG but is only present in S. typhi and S. paratyphi C, therefore far more specific. SSEJ, this target is used to distinguish between S. typhi and S. paratyphi C, however is absent in S. typhi and therefore only useful when screening isolates. Using data from 2016 to identify hotspots of transmission by mapping patients' households to rivers in Blantyre, samples including 1 litre water samples, more swabs, and biofilms were collected. Over an 8-month period, 6 strains of S. typhi were identified from rivers, 1 from a water filtrate, 3 from more swabs, and 2 from biofilms. Using a risk ratio calculation, we determined the positivity rate of trap sample was 44.33 times more likely than the grab samples. Due to mixed culture from these samples, chloramphenicol discs were used to isolate the 6 strains, which were then reconfirmed by PCR and verified using traditional biochemistry, API-20E, serology, O9, O12 and VI, and antimicrobial testing for multi-drug resistance, chloramphenicol, ampicillin, and cotrimoxazole. Thank you for listening, and to my supervisors, collaborators, and funders listed here.
I'm Olumuyiwa Igbalajobi, a postdoctoral research fellow at the Professor Jim Cruster Group at the Michael Smith Laboratory, University of British Columbia, Canada. The title of my flash presentation is Dissecting the Roles of the Proteasome Subunit DSS1 and the capsule-associated protein Cap60 in the ubiquitin proteasome system in Cryptococcus neoformans. Cryptococcus neoformans is a notorious yeast that causes an estimated 300,000 cases of cryptococcal meningoencephalitis per year, resulting in about 200,000 deaths globally, with the greatest occurrence in HIV and AIDS population in sub-Saharan Africa. An impaired proteasome system is a promising area for cancer therapy and the treatment of infectious disease such as tuberculosis and malaria. A major discovery from our published work and preliminary studies shows that the proteasome and the ubiquitin proteasome system play an important role in the regulation of capsule formation in Cryptococcus neoformans. For instance, in Cryptococcus neoformans, treatment with the bipolar disorder drug lithium influences capsule size, and mutants with defect in UPS component have altered lithium susceptibility and capsule size. Therefore, our hypothesis is that the UPS regulatory factors such as DSS1 regulate UPS component and the activity of the proteasome to influence capsule formation and virulence. So far, we've observed that the loss of DSS1 and Cap60 resulted into impaired growth at 37 degrees compared to the wild-type strain. In medium supplemented with cell wall perturbing agents such as Congo Red and SDS, the Cap60 and the DSS1 mutant strain displayed altered growth. Same scenario was observed in medium supplemented with sodium chloride, hydrogen peroxide, as well as sodium nitrate, suggesting a crosstalk between the DSS1 and the Cap60 in stress signaling cascade. Currently, we're using qPCR to analyze the transcript level of Cap60 in the DSS1 mutant as well as the wild-type strain using the capsule-inducing and non-inducing conditions. Ultimately, we hope our findings will present opportunities to discover novel antifungal targets while understanding the role of the ubiquitin proteasome system in fungal virulence. Thank you.
Hello, I'm Nida from Center for DNA Fingerprinting and Diagnostics, India. And before I get into my research, I want to discuss one of the largest threats to food security, global health and development today. That is antimicrobial resistance. Antimicrobial resistance can affect anyone of any age in any country. It considerably increases the duration of hospital stays, medical costs and mortality. A growing number of infections are becoming difficult to treat, such as tuberculosis, typhoid, pneumonia, and the list goes on, as the antibiotic to treat them becomes less effective. So, how can we solve this problem? We can combat antimicrobial resistance through infection control, surveillance, spreading awareness and promotion of rational use of antibiotics, and through basic and applied research that includes everything: understanding of microbial pathogenesis, diagnostics, vaccines, drug discovery, and therapeutics. The fundamental of these research lies in understanding the cellular and molecular processes by studying the structure, function, and mechanism of action of its various components. And this is exactly what I have done. I have studied the function, structure, and mechanism of bacterial endoribonuclease RNA-Z. These studies emphasize it as a potential target for drug discovery and therapeutics, since preferential processing and degradation of RNA by RNA-Z renders it a central player in the homeostasis of the cell, and its function is essential for cell survival. It plays a critical role in pathogenic bacterial virulence. All of RNA-Z homologue, including the ones present in the pathogenic bacterial species, are structurally and functionally very similar, and these homologues are absent in animal or human, thus reducing the risk of any non-targeted effect. So what basically is RNA-Z? RNA-Z is a 1061 amino acid long polypeptide encoded by RNA gene with a catalytic domain in its N-terminal half and a degradosome assembly domain in its C-terminal half. RNA-Z is involved in global mRNA turnover, maturation of tRNA and rRNAs, and although it's an essential endoribonuclease, its C-terminal half is dispensable for viability. Here is a linear representation of the functionally important residues of RNA-Z. RNA-Z has a 5' sensor pocket with a critical residue R169 that is allosterically regulated by 5' end of RNA, and 5' monophosphate is associated with maximal activity. It also has an active site pocket that includes residues D303 and D346. All these residues are present in the N-terminal half and are retained in all the truncations, such as 529, 493, 438, 418, and 395 delta, where there is complete absence of small domain along with the zinc link that contributes in oligomerization. With this background, I am going to start my talk, Two Halves, Two Tails of RNA-Z. In the first part, I will talk about cross-subunit catalysis and intragenic complementation. Then I will discuss a new phenomenon labeled as recessive resurrection, and lastly, aggregate or condensate dependent toxicity. One assay that I have used throughout to test for viability is the blue-white assay. The phenotype of viability or lethality of RNA-Z was investigated through a RNA plus lag-Z plus unstable single copy shelter plasmid that gives perceptible blue color on X-gal supplemented plates to the colonies that are chromosomally delta lag. So when RNA allele is lethal, then even without the selection marker, only blue colonies will be observed on the plates, as cells that have lost the shelter plasmid can't grow. Whereas when RNA allele is viable, then the plasmid-free cells are also observed as white colonies on the plates. Using this assay, different RNA-Z truncation variants were examined, and all the RNA truncation derivatives, including 395 delta, are able to confer viability. These truncation variants were further studied using RNA-lag assay. The rationale for this assay is RNA-Z transcription is autoregulated. Since RNA-Z cleaves within its own 5' untranslated region to initiate degradation of RNA-mRNA. Hence, RNA-lag expression is inversely related to the total cellular RNA-Z activity. The result of chromosomally deleted RNA lag strain carrying truncation variants on a single copy number plasmid to confer viability shows that the in vivo catalytic activity is progressively reduced with increasing length of RNA truncation. The RNA-lag assay in RNA plus strain has shown that most severely truncated RNA derivatives are more dominant negative in vivo, indicative of oligomerization with and subunit poisoning of wild-type RNA-Z. This includes 395 delta. Again, with the help of the earlier mentioned blue-white assay, the ability of different RNA-Z constructs to confer viability to a del RNA strain was tested. Based on the assay, visible white colonies indicating viability appeared on RNA plus 529 delta, 493 delta, and 395 delta, while 5' sensor mutation R169Q was lethal in RNA-Z delta CTH, and active site mutation D03A and D346A are lethal even in full-length RNA-Z. Interestingly, there is a restoration of viability upon co-expression of these pairs of lethal RNA allele, demonstrating in vivo intragenic complementation. As can be seen in the panels, that viability is conferred when expressed together from two different polypeptides, but not from the same polypeptide. Even RNA-Z 395 delta polypeptide with individually lethal sensor or catalytic mutations confer viability when co-expressed. This restoration of viability upon co-expression of pairs of lethal RNA allele was also reconstituted in vitro. RNase truncation derivatives were purified and used in RNase cleavage assay together with a 13-mer 3' fluorescein labeled RNA substrate. This releases a 5-mer cleavage product which was observed along with the temporal increase in product formation upon combined addition of the pair of polypeptide, each individually inactive. Hence, the findings so far suggest that 395 delta, lacking all known sequences for oligomerization, does oligomerizes, and its RNA cleavage mechanism is apparently through 5' sensing by one subunit and endonuclease cleavage by another. But a cross-subunit sensing and cleavage mechanism is incompatible with a model of monomer catalytic activity. So, are they forming dimers? If yes, then how? Could RNA binding play a role in dimer assembly? To check this, I have done size exclusion chromatography with a 13-mer RNA substrate having 2' methoxy groups on ribose moieties so it can bind to RNase without getting cleaved. Control proteins showed no difference with the absence of RNA, green line, and presence of it, the black line. But 395 delta, which showed no effect with change in salt concentration, does show different elution pattern with different amounts of RNA. Both RNase 395 delta and 493 delta variants showed slower elution profile in the absence of non-cleavable RNA, which is interpreted as monomer, than in its presence, which is interpreted as dimer. Conclusion from this part of the work is that intragenic complementation occurs by RNA-facilitated oligomerization and cross-subunit catalysis between a pair of RNase 395 delta polypeptides. This elucidation of the structure and catalytic property of RNase is important for the development of effective antimicrobials targeting RNase, which emphasizes it as a potential target in drug discovery and antibiotic resistance studies. With this, we move on to another interesting part of the work, which started as the study of the effects of lethal mutations on RNase activity. The 5' sensor and active site mutation confer dominant negativity when C-terminal half is deleted. This was demonstrated by the RNA lag assay in both chromosomal 493 delta and RNA plus, carrying the indicated RNase variants. The red line in these figures represent RNA lag value of RNA strain with vector, and as RNA lag expression is inversely related to RNase activity, so any bar higher than the red line means lesser activity than the wild type, indicative of dominant negativity. An expected outcome of oligomerization of RNase with lethal mutations. To our surprise, this was not the case with full-length RNase. The RNA lag assay in chromosomal RNA plus RNA lag strain have shown that full-length heterotetramer composed of a mixture of wild-type and recessive lethal active site mutant of RNase exhibited identical activity in vivo as the wild-type RNase homotetramer itself, a discovery of a novel phenomena labeled as recessive resurrection. Comparatively, the control lag fusion ProU-lag was unaffected by the overproduction of wild type or the mutant RNase, indicating that observed effects were specific to RNA lag. The phenomena of recessive resurrection was also observed with the double mutation in both chromosomal RNA lag strain RNA plus or 493 delta. As recessive resurrection was an entirely new discovery, it was necessary to exclude all the alternative possibilities. Accordingly, several experiments were conducted as tests for the authenticity of recessive resurrection, such as confirmation of mutation in the gene, equivalent expression of all the polypeptide, no difference in the growth rates between all the ASCA plasmid, and determination of 5' UTR levels for mRNA expressed from chromosomal RNA plus locus by RT-qPCR. Interestingly, the same 5' sensor and active site mutations that show the phenomena of recessive resurrection in full length confer dominant negativity when C-terminal half is deleted. This suggests that the CTH is involved in mediating the phenomenon. Accordingly, the proposed model is that CTH mediates a rate-determining initial step in catalysis, which may perhaps be the binding and channeling of RNA substrate for NTH endonucleolytic action. Conclusion of this part is a new genetic phenomena was discovered which can help in identification of previously unidentified genetic defects due to single copy gene dosage masking the phenotype in a diploid organism. This novel phenomena should be actively looked after in all species, especially human. One approach to identify them in diploid organism may be to focus on genes that are known to display haploinsufficiency phenotypes, and then to identify missense mutations in them which lead to a loss of function in the homozygote and yet create no haplosufficiency defect in the heterozygous individual. Moving further into the C-terminal half and last part of my work, this is the linear representation of the CTH of RNA-Z. The non-catalytic CTH of RNA-Z harbors RNA binding domain RBD, membrane targeting sequence MTS, and serves as scaffold for other proteins of degradosome PNPase, enolase, and RHLB. The characteristic feature of being unstructured is highly conserved, implying that the disordered region could be having a significant physiological role. The last part of my talk deals with finding an explanation of RNA toxicity in E. coli. Toxicity of RNA-Z and its variants were tested by expressing it in a wild-type strain. The plasmid expressing RNA-Z and its variants were IPTG-regulated ASCA plasmid. This means that an increase in the IPTG concentration would result in an increased gene expression. Therefore, an inverse relationship is expected between IPTG concentration at which absence of colonies are observed and the level of toxicity. As can be observed in the results, the vector is not toxic at any concentration, and wild-type RNA-Z is toxic at 50 micromolar. This toxicity appears to be decreasing with truncation of CTH as observed for 529, 493, and 395 delta. Surprisingly, toxicity of RNA-Z is independent of its activity, as even catalytically inactive polypeptides are toxic. This toxicity also decreases with CTH truncation. The CTH by itself also confers toxicity at 50 micromolar IPTG, which is equivalent to the toxicity conferred by full-length RNA-Z. This made it clear that the C-terminal half is responsible for toxicity, and so the components of CTH were further tested. Degradosome does not appear to play any role in RNA-Z toxicity, whereas membrane binding might have a role. Toxicity could also be due to aggregation in the bacterial cytoplasm, similar to amyloidogenic proteins in eukaryotic cell. Such property of RNA-Z is also consistent with the high propensity of intrinsically disordered region IDR found in CTH. Further, microscopy studies with IBPA YFP fluorescent fusion proteins were done, and it was noticed that the membrane localized polar foci was observed only in strains expressing CTH of RNA-Z, either in truncation or full length. Binding of IBPA and foci formation does indicate aggregate or condensate formations. Further experiments are being done in this direction. Our current model is that the aggregation or condensate formation of RNA-Z occurs due to multivalent interactions of IDRs and RNA binding by RBD, and the toxicity is the result of an overall increase in the local concentration of these formations due to membrane localization of E. coli RNase. These results will have further implications as they suggest a mechanism similar to the formation of aggregate, prion, amyloid or phase-separated liquid or gel-like condensate. These are already known to be the cause of many diseases and disorders in animals and humans. Studying these phenomena in bacteria will provide insight into the fundamental principle of these processes, which will further aid in diagnostic and medical research. With this, I end my talk. Thank you for joining me.
Hello, I'm Kelly and I'm going to give you a super quick presentation on part of my PhD research, looking into the combination of biocides and quantum sensing inhibitors together to prevent uropathogenic E. coli infections. Catheter-associated urinary tract infections, CAUTI, are a significant problem to the healthcare systems across the globe. Many people with a long-term catheter in place develop an infection, and this can lead to complications, in the worst cases septicemia and death. Uropathogenic E. coli, UPEC, are a major causative pathogen of CAUTI. UPEC form biofilms on the catheter surface, and many strains harbour antibiotic resistance, which makes treating an infection a significant challenge. Biocide-impregnated catheters have been developed to prevent CAUTI. However, these have been seen to have short-lived efficacy, and there's building evidence of biocide resistance mechanisms arising in bacteria. The high concentration of biocides needed to eradicate a biofilm also has potential cytotoxicity. To overcome the challenges of using biocides alone, I'm combining them with quantum sensing inhibitors. UPEC communicate through the autoinducer 2 mechanism of quantum sensing, and there's been observed correlation between AI2 signalling and biofilm formation. Quantum sensing inhibitors, like cinnamaldehyde and furanone, which I'm using, interrupt the AI2 signalling pathways. I'm combining these with the biocides and investigating the possibility of a synergistic interaction, which means there will be a lower concentration of both agent required to have the same impact as a single agent alone. To test this, I performed checkerboard synergy assays on UPEC strains to determine the efficacy of the biocides with the quantum sensing inhibitors. These graphs represent the fractional concentration index values obtained from challenging UPEC strain EC958 with the biocides and quantum sensing inhibitors. A value of below 0.5 represents a synergistic interaction. As you can see, a synergistic interaction was observed at the biofilm eradication concentrations for PHMB, BAC and silver nitrate in combination with both of the quantum sensing inhibitors. A similar pattern was observed in other strains that I tested. Using this data, I'm looking to combine the effective biocides and quantum sensing inhibitors into a sol gel coating, and this will be applied to a urinary catheter surface. Ongoing work into the cytotoxicity of the combinations, elution of the agents from the coating, and then into the validation of these coatings is ongoing. Just want to say thanks to my supervisory team, led by Dr. Sarah Forbes, and to the BMRC for funding my research.
Hello and Namaste, I am Sujata Baidya, Medical Laboratory Technologist and currently a postgraduate student at Institute of Medicine, Tribhuvan University. Today I am going to talk on association of biofilm formation and antimicrobial resistance in Pseudomonas aeruginosa, isolated in ventilator-associated pneumonia at a tertiary care center in Nepal. Biofilms are said to be associated with nearly 50% of the nosocomial infections. Their presence in device-associated infection has made the treatment process even more complicated, causing chronic and recurrent infections. Biofilm in endotracheal tube has paved various ways of antimicrobial resistance, causing the biofilm-associated organisms to be about 10 to 1,000 times more resistant than their planktonic forms. FAB has high incidence in Southeast Asian countries. In developing countries like Nepal, where the health infrastructures are limited, the condition can be even worse. Moreover, biofilm associated with these diseases can increase the treatment cost. The objective of our study was to unravel the relation of biofilm formation with antimicrobial resistance in Pseudomonas aeruginosa. It was a 6-month study which was conducted at Tribhuvan University Teaching Hospital, Kathmandu. A total of 70 lower respiratory tract samples were taken from patients under mechanical ventilations and processed as per standard guidelines. Biofilm was detected by microtiter plate method. In our study, a total of 71 organisms were isolated. 31% of them were Pseudomonas aeruginosa, and 63.6% were biofilm producers, while 59.1% were MDR. Among the isolates which were resistant to individual antimicrobial agents, bulk were biofilm producers. It also reveals a statistically significant relation between biofilm formation and piperacillin tazobactam resistance. So our study concludes that biofilm formation further aggravates the thriving drug resistance in VAP. But early biofilm detection can help to reduce the mortality and morbidity associated with it by modulating the treatment strategies. We are really grateful to the entire hospital staff and the patients who contributed to this work. Thank you all for your time. Lastly, I am grateful to the Society for Applied Microbiology for providing us with this virtual platform to share our work, and I will be very happy to communicate on any queries in the future with the contact I provided here. Thank you.
Hi everybody, I'm Diana Andrade from the Research Unit for Comparative Microbiome Analysis at Helmholtz Center in Munich. I would like to present my work on soil microbiome response to climate change and agricultural management in montane grasslands in the frame of our SUSALS project. Montane or pre-alpine grasslands are important for water retention, nutrient storage, biodiversity conservation, fodder production, as well as for human recreation and tourism. However, pre-alpine grasslands in Europe are exposed to increasing temperatures twice as fast as the global average, accompanied by increased land use intensity to improve productivity. Therefore, we investigate the combined effects of climate change and land use intensity, which is needed to further implement agricultural management strategies and avoid soil nutrient losses. We present results from our study performed in the pre-alpine region southern Germany, represented here along an altitudinal gradient where the dominant nitrogen input is the cattle slurry fertilization, which is, however, extensively applied only at the higher altitudes. We combine an extensive or intensive management with a space for time climate change experiment, which was based on the translocation of intact plant soil mesocombs down this elevational gradient to induce warming of 1 and 3 degrees Celsius. After the first and second year of exposure, we quantify respectively by qPCR the main functional groups of microbes involved in the nitrogen and phosphorus cycle and measure soil plant properties in these mesocosms. Our data indicate significant interactions between climate change and management. For example, if we look at the nitrogen cycle, N-mineralizing bacteria increased in abundance under extensive management, while under the intensive treatment, the microbial biomass was reduced up to 50%, accompanied by lower abundance of ammonia-oxidizing and nitrogen-fixing bacteria. In contrast, P-mineralizing bacteria and bacteria with strong P-uptake systems decreased by an extensive management, while under the intensive treatment, the P-mineralization was increased. Concluding, we can say that N and P turnover is more influenced under the stronger climate change effects. Interacting effects of climate change and agricultural management can indeed affect microbial community at the short term, which therefore will have long-term consequences for the soil nutrient stoichiometry, and these are just the definition of microbial indicators for the different management under climate change effects in favor of soil health, productivity, and thus in the human well-being. I thank you for your attention and will be happy to answer your questions. See you!
Thanks to Society for Applied Microbiology for this conference opportunity to present on natural microbial communities from Niger Delta facilitate anaerobic benzene mineralization under iron-reducing and methanogenic conditions. Niger Delta is one of the world's most important wetlands. However, oil spills have damaged the environment and devastated means of livelihoods and impaired ecological services. The goal of the study was to elucidate the potential of Niger Delta sediment for anaerobic hydrocarbon natural attenuation processes using benzene as a model compound. Specifically, we assessed the metabolization and mineralization with 13C labeled benzene and characterized the microbial communities thereof. Sediment from Ogoni, 4 different locations in Ogoni, showed high biodiversity. Focus on canned sediment shows relatively high abundances of gamma proteobacteria, Bacteroidea, and halophagels. The methanogic phylotypes were more of Methanosacina, Methanocella, and Methanobacterium. The Kano regional sediment was transferred to platonic cultures to assess benzene mineralization under sulfate reducing, iron reducing with 3 different ions and the methanogenic condition. The result shows that benzene was mineralized under iron reducing conditions at your right panel and methanogenic conditions, the upper panel, while no mineralization took place under sulfate reducing conditions. Cultures amended with iron 3 ores that showed highest mineralization rates up to 2000, and cultures amended with iron 3 nitrotriacetic acid showed we enriched in 13C methane. Microbial community structure generally for the bacteria indicated affiliated to beta proteobacteria, the sulfomonadels, and the Gnavibacteria, while the methanogenic phylotypes were more of the Methanosacina and the Methanotris. The enriched mineralizing communities were diverse, as we can see, and contain more than a single benzene degrader, which is opportunity for further study on this. And we conclude that benzene was slowly mineralized under 2 conditions, and phylogenetically different organisms mineralizing benzene in the enrichment comes from Niger Delta. This underpins the importance of microbial ecosystem services such as contaminated degradation. Thank you for your
Hi, my name is Hollie Shaw. I'm a student at the University of Sheffield at the Dental School, and I am studying the effects of low-intensity ultrasound on Staph aureus biofilms. If you're a bit squeamish, there is a picture of a wound coming up, so if you don't like that, please be warned. Why are we interested in these biofilms? Well, they're often found in chronic skin infections, so 60% of chronic wounds have a biofilm present, compared to only 6% of acute wounds. And these chronic infections are slow to heal. They often need repeated or extended use of antibiotics, which is not very good antibiotic stewardship, and there's also increased risk of complications because these infections can penetrate deeper, become systemic infections or bone infections, which require more drastic treatment options. So why are they more difficult to treat? There's a number of different reasons. So one reason is there is poor diffusion of the antibiotic through the biofilm. So these antibiotics can't get to the targets of the cells because of the extracellular matrix. There's also enzymes present within the extracellular matrix, which are there to degrade antibiotics, so they don't reach the target either because they are inactivated before they even get to the cell. And then there's also the presence of antibiotic-tolerant persister cells, and these are alive but metabolically inactive cells within the biofilm, which do not exhibit any of the metabolic targets which antibiotics normally target, such as protein synthesis or wall synthesis, because they are sleeping. So that brings me to my project, and there's evidence to suggest that using ultrasound can increase bacteria sensitivity to antibiotics. So that is my project. Does it make biofilms more sensitive? How does it make it more sensitive? Does it work in tissue engineered models? So this is the machine I used to ultrasound my biofilms. It is a prototype of an Exogen machine, which they don't look like this anymore, but they are used in fracture clinics. So they are used by clinicians and they are well tolerated by patients. This is some preliminary data which I have, which shows that biofilms at various stages of maturity do exhibit increased sensitivity to gentamicin when treated with low intense pulse ultrasound. And there may be evidence to suggest that it may be changes to structure and permeability of these biofilms, which causes the increased sensitivity that we see. So the wider applications and benefits of this project is we're hoping that it would translate into clinical use, and this would then improve antibiotic stewardship. This would then in turn reduce the risk of further antibiotic resistance developing. It would also hopefully improve the treatment times for these patients with these wounds, and they would heal quicker, reducing their risk of complications and ultimately improving their quality of life, which is the most important thing. So thank you very much for watching and listening, and find me on Twitter at Hollie, and let's chat.
Hi everyone, my name is Erin Magee, and today I'm going to be giving you an overview of my research into the development and use of 3D printed smart wound sensors for infection monitoring and biofilm development. So first, to give you an introduction into chronic wounds. So they have been described as a silent epidemic because the true impact on both the financial and healthcare basis is often masked by the comorbidities associated with it. The top three being diabetes, obesity and cardiovascular disease. In addition to this, they predominantly affect the elderly population, which represents the fastest growing age group and is expected to double in proportion by 2050. So you can appreciate that this is going to be an even bigger problem in years to come. So wound healing is a very complex process, but it can be divided into four main stages, these being coagulation, inflammation, proliferation, and finally remodeling with the formation of scar tissue. In normal wound healing, we would see an elegant progression through these four stages in a timely manner, usually within 3 months, and this would lead to full restoration of the skin. However, chronic wounds are typically confined to the inflammatory phase. This can persist for a number of months or possibly even years, and in some cases may necessitate amputation. So it can have a huge impact on the patient's quality of life. So biofilms are actually present in up to 80% of all chronic wounds, and as we know, they lead to significant treatment difficulties in terms of heightened antibiotic tolerance and the formation of persister cells. One of the top methods for treating chronic wounds is surgical debridement, but this is often unsuccessful as it will miss bacteria inhabiting the deep layers of the skin, which can then propagate infection once again, and so you're back to square one. So successful treatment really relies on early detection. It's far easier to treat a biofilm in the early stages than when it is robust and is spanning both the superficial and the deep layers of the tissue, and that's what this work focuses on. So it's developing a sensor that provides 24-hour monitoring of the wound environment, flags early signs of infection and improves treatment outcome overall. So the indicator we have developed is a 3D printed carbon dioxide indicator. The reason we chose carbon dioxide as an indicator of infection is that it is produced by all bacteria as part of normal respiration and metabolism. So the CO2 indicator contains a dye called xylanol blue, which is essentially a pH indicator. So under normal atmospheric conditions, where CO2 is at about 0.04%,
xylanol blue exists in its deprotonated blue form. As the bacteria produce CO2, the CO2 mixes with water in the air to form carbonic acid, which protonates the dye. So as we produce a bit of CO2, then we have about a 50 to 50% portion of protonated to unprotonated dye, which gives us a green intermediate. And then as the percentage of CO2 increases even more, the dye becomes fully protonated and we get this yellow. So you get a nice transition from blue to green to yellow. The colour change is reversible and it is entirely dependent on local CO2 concentrations. The film itself is ion impermeable, so it is not affected by any ions in the wound exudate. And the way the film is made, it has high permeability to CO2, but low permeability to any other interfering gases. So it is very specific for carbon dioxide production. So just to give you a brief overview of how we actually make the indicator. So the dye is mixed with hydrophilic silica, a quaternary ammonium base and water, and this is then spray dried to form a powder pigment. This pigment is then blended with LDPE and fed into an extruder to form pellets. The pellets are repelletized 3 times just to ensure that the pigment is thoroughly mixed throughout. And then these master batch pellets are fed into the extruder to form a 3D printable 5% weight pigmented film. And of course then this filament is 3D printed to form the square of 1 centimeter diameter and 50 micrometers thick. And you can see the images of that on the right of the screen here. And down below you can see the transition of the colour from 0% CO2 all the way up to 100. And you can see the nice transition from blue to yellow. And all of this work was conducted at 30 degrees because that's the temperature of the skin in the areas of the body that we're interested with regarding chronic wounds. So we tested the sensor using an ex vivo porcine skin model. The reason we use porcine skin is because it is the best substitute for human skin. So first of all, you shave the hair off the skin and remove any excess tissue. We then disinfect with 70% ethanol to remove any commensals, and then we emulate a chronic wound by making a burn wound, as you can see here. This is then inoculated with 20 microliters of whichever bacteria you're using and then sealed in the wound dressing. So I'll just quickly run through the setup of this. So we have a supporting PET plastic at the bottom, an absorbent pad to which the porcine skin is put on top of. We then have 2 layers of lint dressing, the indicator film, and then a breathable Tegaderm dressing on top to hold it all in place. So then we take an image of the film every hour for our RGB colour analysis. So moving on to some experiments. So I focused on Pseudomonas aeruginosa infection because it is a key infection protagonist in the wound bed, and it loves to form biofilms, which makes it the perfect candidate to work with. So we used a clinical isolate of Pseudomonas, this one here, PA10548, and I inoculated the skin with 3 different concentrations: high 10 to the 6, medium 10 to the 4, and low at 10 to the 2. And as you follow the indicator response over time, you can see that the 10 to the 6 changes first, followed by 10 to the 4, and then 10 to the 2, as you would expect. We can then convert this image into a graph where the y-axis here stands for A apparent, which is the apparent absorbance, which is essentially just the colour change in a numerical term. And you can see that it transitions from blue through green down to yellow, and the time gaps in between obviously displaying that there's a difference in time between the inoculum and the sensor response. And then finally, here's a parallel experiment for the viable counts of the bacteria in the wound dressing over time at the different inoculated concentrations. And the grey line here just shows the limit of detection of viable counts. So if we go back in and look at this in greater detail, so the red circles here highlight the colour at which the sensor is starting to change. So it's kind of like a blue, green, teal colour. And we're interested in this because this is obviously the point that there's enough bacteria to start tripping the sensor. So for 10 to the 6, this is 11 hours, 10 to the 4, it's 14, and 10 to the 2, it's 23. Now, if we go to our sensor colour change graph, draw a line at 0.8, which is where we would typically find that teal colour, and draw down the x-axis, and we see that that matches up really well. 11 hours, just over 14, and 23. Now the important bit is when we transfer that information to the viable counts. And once we put our times on and go up to the growth curve across the y-axis, we can see that the sensor seems to trip in and around 10 to the 6 CFU per mil, which is really important because that would be a clinical sign of a wound infection. We are also able to alter the sensitivity of the film by playing around with the base concentrations, so it is possible, and we are looking into making the sensor even more sensitive, possibly tripping upwards of about 10 to the 4 bacteria. So we have also correlated the exact colour to the percentage of CO2 using FTIR, as you can see in this diagram here. So we had a custom-made IR gas cell with our porcine skin inoculated with Pseudomonas and the CO2 sensor fitted inside. So you can see the CO2 peak increasing with each hour that passes and the corresponding colour change. From this, we can then calculate the percentage CO2 for a given time, and then we can show that this is actually linearly correlated to the exact sensor colour, which is really important because it possibly adds a diagnostic aspect to the film. Of course, it is important to monitor the onset of infection, but it would also be really great to monitor the efficacy of treatment. So for this experiment, I grew the Pseudomonas aeruginosa biofilms for 24 hours on pigskin and did 2 treatment options. So the first was with a phage cocktail that I knew was only going to get about a 2 log reduction in bacteria, so I knew it wasn't going to completely eradicate the infection. And then gentamicin at a concentration of 64 micrograms per mil. From previous experiments, I had worked out that this was the minimum concentration of gentamicin required to eradicate the biofilm grown in porcine skin. So they were grown, biofilm was grown overnight, then treated overnight, and then washed in PBS to remove any planktonic cells, and then transferred to the wind dressing with the indicator for monitoring. So first we'll look at the phage cocktail. So as you can see, the untreated changed colour immediately because there's a high number of bacteria there producing CO2, which trips the indicator, and this is confirmed by the counts on the right hand graph. Then if we go to the treated explants, you can see that they remained blue for about 3 to 4 hours, but after that then they eventually did transition to yellow because there was bacteria on the skin. And when we look over to the graph, we can see that there is as much as 10 to the 6 bacteria, which completely explains why the sensor responded to this. So the treatment was unsuccessful. Now moving on to the gentamicin experiment. Again, the untreated changed colour very quickly because there was a high number of bacteria on the skin. But when we treated with 64 micrograms per mil, you can see that the sensor remained blue for the entirety of the experiment. So there was no bacteria there, no CO2 produced, so the sensor didn't change colour. And this was confirmed by the counts on the right hand side. So here we have a successful treatment. So this is really encouraging data for us to show that we can use it to detect infection but also monitor the efficacy of treatment. So just to conclude, the CO2 sensing film that we made housing xylanol blue dye is a very strong candidate for effective 24-hour real-time analysis of early infection development. It has a striking colour change of blue to yellow that is easily visualised with the naked eye. We're also able to linearly correlate the percentage of CO2 to the exact colour change, which adds a promising diagnostic aspect to the film, and the sensor can be used to monitor treatment efficacy over time. First of all, I'd just like to thank you all for listening and SFAM for giving me the opportunity to give this presentation. I'd like to acknowledge Dr. Jilly Yusuf, who I work with in the School of Chemistry at Queen's. He helps immensely with the sensor and the chemistry aspect. My supervisors, Professor Brenda Gilmore and Professor Andrew Mills, and of course the lovely people that I work with, and the funding bodies, DFE and UKRI. And if you have any questions or want to get in contact with me, I've left my contact details at the bottom. Thank you very much.
Hi, I'm Alice, and I'm going to talk about the bug that makes our drugs. So therapeutic proteins, things like enzymes, hormones like insulin, antibodies and vaccines, are often produced in the bacterium E. coli. And quite a lot of these are secreted to the periplasm, the area between the inner and outer membrane, to improve protein folding, which is what I'm interested in. However, this can be challenging, and you can often get quite low yields and overwhelm the cell. So it often requires a lot of time and money and trial and error in order to optimize this. So as part of my project, I want to understand how E. coli responds to secretion of therapeutic proteins. And I'm doing this using transcriptomics and proteomics to compare E. coli expressing a model therapeutic protein, an SCFB antibody fragment in the cytoplasm, to E. coli that secretes this protein to the periplasm as we've added a signal peptide. And we hope to use this data to then engineer strains to improve protein secretion. So secretion of our model protein had a significant effect on both gene and protein expression. And there were some key areas affected by this that we were particularly interested in and would like to modify in the future. These involve the envelope stress response, which is known to respond to unfolded periplasmic and membrane proteins, expression of periplasmic chaperones, which fold periplasmic proteins, cytoplasmic chaperones, which can play an important role in secretion by guiding proteins to be secreted or sequestering them. We saw that secretion led to a decrease in expression of the sex secretion system, which is used to secrete our protein as well as lots of other therapeutic proteins. And we also saw that this secretion system was being potentially degraded, which we'd also like to modify. And so we've identified 13 genes in our proteomics and transcriptomics that we've knocked out and overexpressed, and are now in the process of testing whether they improve production of our SCFV as well as other proteins, and test the effect of these on growth. So thank you for listening. If you have any questions or comments, feel free to contact me.
Hi, my name is Marta Filipa Simões, and I'm here today to tell you a little bit about metallic nanoparticles as potential biosignatures of life. As we know, metallic nanoparticles have a wide range of applications. They can be produced by different methods, physical, chemical, and biogenic. Regarding the biogenic productions, we know nowadays that members from all the branches of the tree of life have been reported as being producers of metallic nanoparticles. To show you how easy and simple is this production, I'm going to give you an example of filamentous fungi. So if you consider a petri dish of filamentous fungi, and if we cut little plugs from grown colonies and put them into appropriate media and allow them to grow a little bit further, if we then remove this growing biomass into sterile water and allow this growing biomass to release all the fungal metabolites into this water, we are then left with a supernatant if we remove all the biomass. And this supernatant would be filled with the fungal metabolites. If we add to the supernatant a chemical precursor, we will have the formation of metallic nanoparticles, and this will be noticeable by an immediate change in color. Of course, different chemical precursors will be used to obtain different metallic nanoparticles. If we consider the same method, but instead of using a biological substance, a biogenic biomass, the fungal biomass, we use a sample, let's say soil from Mars, and if we mix the soil into sterile water and then add to this mixture a chemical precursor, if we do have formation of metallic nanoparticles, then we can assume that the initial solution, which in this case would be the soil from Mars, will likely have biological substances that allow for the synthesis of the metallic nanoparticles. Taking also into consideration the definition published in 2008 that says that a potential biosignature is a feature that is consistent with biological processes, and when found it challenges the researcher to attribute it to either inanimate or biological processes, then we can consider that the biogenic synthesis of metallic nanoparticles is a potential biosignature. This would be a simple, fast, cheap way to do a screening of life, and it wouldn't need any advanced or specialized or dedicated equipment or personnel. Furthermore, it would be easy to perform and implement either on Earth or with some adaptations in situ. Of course, there are still some challenges regarding this. We need additional research, we need to define the limits of detection of the metallic nanoparticles, and we need to better understand their formation. And of course, any formation of metallic nanoparticles when doing the screening would still need confirmation that it came from a biological origin. So we do need to develop a method to distinguish biogenic from non-biogenic metallic nanoparticles. Thank you very much. This was shortly what I wanted to tell you about. If you want to discuss this a little bit further, please contact me via email or Twitter. Thank you very much. Enjoy the rest of the conference. Bye.
Good day to you. My name is Akindolire M.A. I am a postdoctoral fellow at ARC, Agricultural Research Council, South Africa. And the topic of my talk is isolation of psychotrophs from psychrophilic anaerobic batch culture. Cold-adapted microorganisms, including both cold-loving saprophytes and cold-tolerant psychotrophs, are widely distributed in nature and exist in deep oceans, glacier, and polar ice, where they contribute to biomass production, nutrient cycling, and methane production from the degradation of organic compounds. In order to survive and proliferate in cold environments, psychrophilic microbes have evolved a complex array of adaptive mechanisms in maintaining membrane fluidity, synthesizing cold acclimation and cold active proteins, and also in possessing various freeze tolerance strategies that have enabled them to overcome challenges presented by life in the cold. Consequently, recent research has focused on gaining a better understanding of these microbial processes and cold adaptation mechanisms with tremendous potential for biotechnological applications, particularly anaerobic digestion. Anaerobic digestion is a well-established technology for the production of renewable energy and for valorization of organic waste. However, process failure results from a drop in temperatures during cold climatic conditions. It is therefore necessary to establish performance improvement strategies during cold conditions in order to offset the effects of low temperatures on process efficiency. Bioaugmentation, or the inoculation of AD systems with specific strains or mixed culture of microbes that possess improved degradation ability, is a promising approach to enhance digestion efficiency and solve the problems associated with low temperature AD. Therefore, in an attempt to get an insight into the important microbial processes occurring at low temperatures during psychrophilic AD, and investigate cold active microbes, this present study aimed to isolate cold adapted psychotrophs from psychrophilic anaerobic digestion. At the time of presenting, 20 saccharotolerant microbes were isolated from saccharophilic bark culture, as shown on this image. And this isolate with further characterization could be utilized to augment saccharophilic anaerobic digestion. I'd like to appreciate the National Research Foundation of South Africa and Agricultural Research Council South Africa for funding this study.
My name is T.R. Duche, presenting a paper on in vitro screening of bacterial isolates from traditional fermented sorghum products for probiotic use. These bacteria were isolated from fermented sorghum products and screened for their ability to survive gastrointestinal acidity at different pH using simulated gastric juice. They were also tested for biotolerance and antimicrobial activity. The results here provide the basis for further screening of isolates from these products to be considered for probiotic use. Most of these fermented beverages from Nigeria are made from sorghum. They have a high content of bacteria with large biodiversity. It is on this ground that we decided to explore the microflora of these beverages, focusing on the beneficial ones for probiotic use. They were screened using different methods such as acid tolerance, biotolerance, and antimicrobial activity. The results here represent that of the survival ability in pH1, pH2, and pH2. Most of them were able to survive up to 4 hours of incubation, and some even survive up to 12 hours after incubation. Figure 4 and 5 is that of the ability to survive in 1% and 2% biosolutions, respectively. Table 1 is the antimicrobial activity of these isolates on selected pathogens by overlay method, while Table 2 is the antimicrobial activity of the isolates on selected pathogens by agar-well diffusion. Table 3 is the co-culture interference of potential probiotic with Salmonella species, while Table 4 is the co-culture interference of the isolate with E. coli. Plate 1 and 2 is showing various zones of inhibition of this isolate on Salmonella and Shigella flexneri. 29% of these isolates survive in simulated gastric juice up to 12 hours of incubation. The result of the agar-well diffusion is an indication that they can be used against pathogens implicated in food-related infections. This fact has also been established by other authors who reported various degrees of antimicrobial activities of lactic acid bacteria and different relevant food-related pathogens. Our findings will serve to promote the role of probiotic bacteria from fermented sorghum beverages to be utilized in the control of food-related infections. Our limitations in this study were that we could not demonstrate the latent survival of this potential probiotic culture during gastrointestinal passage. We could not also screen them for the quality and quantity of antimicrobial substances produced. We suggest further studies for this. We have selected references here to back up our findings. Thanks for your audience.
Can we turn microbes into oil factories, and why does it matter? DHA, POA, and VA are unsaturated fatty acids that are beneficial to human health, including reducing the risk of cardiovascular disease, diabetes, and neurodegradation. Due to the high demand, more sustainable sources are in need. Orontirocitrum and eukaryotic microbe from the ocean may help this, because it accumulates large amounts of lipid with high proportions of unsaturated fatty acids. To turn it into competitive production factory, knowledge of the enzymes that synthesize these fatty acids is crucial. Orontirocitrum species T66 can make DHA, POA, and VA. In a standard fatty acid synthesis pathway, palmitic acid can be desaturated to POA and then elongated to VA. In T66 genome, there is a C16 elongase, but no delta 9 desaturase. This raises the question, how do POA and VA synthesized in T66? At the same time, there is a delta 12 desaturase death in T66 genome, which can desaturate oleic acid into linoleic acid. However, both fatty acids are not produced by T66. Since death is also highly upregulated when the cells actively accumulate lipid, this made we puzzled that what is the function of death? To find the answers, we utilized another species Orantilonchistrum libmacinum SR21. What it is different to T66 is that death is absent in SR21 genome. It also does not produce POA and VA. After overcoming challenging genome engineering procedures, I created a strain expressing death in SR21, which turned out to produce POA and VA. These exciting results showed that death is more likely to be functioned as a delta 9 desaturase than a delta 12 desaturase, which is different to the previous sequence homology. And this information will be beneficial, valuable in metabolic engineering for microbe fatty acid production.
Hello everyone. Before I start my presentation, let me introduce myself first. My name is Hamida Ida. I'm a master's student from Department of Biotechnology, Graduate School of Universitas Gadjah Mada, Indonesia. So, the research that I will present today is under the title Effect of Glucomannan from Porang or Amorphilus onchopilus on gut microbiota of overweight and obese people. This research was done by me and my team in Rispro Glucomannan Porang research team that's sponsored by Indonesia Endowment Fund for Education. So, first of all, I will talk about porang tuber itself or Amorphilus onchopilus, and then I will continue to talk about glucomannan from porang. And the last, I will talk about the effect of glucomannan from porang itself on gut microbiota of overweight and obese people. Okay. This is our research background. This is what we call porang tuber or Amorphilus onchopilus. Amorphilus onchopilus is a local tuber native to Indonesia and easy to cultivate. The production of porang is quietly high in Indonesia. It can reach thousands of tons every year. Porang tuber itself never consumed by our Indonesian people directly because the high content of oxalic acid. The oxalic acid from porang can give you itching sensation when you eat them. Porang also known to have tremendous potential due to its active ingredient in the form of glucomannan. The glucomannan in the porang is quietly high. It can reach more than 70% due to Watani et al. At 2020, depending on its extraction method and then time of harvesting and also the age of porang tuber itself. Glucomannan also known to have potential in prebiotic as a prebiotic activity because glucomannan has beta-1 for glycosidic bond which cannot be digested by human digestive enzyme. So, As a consequence, glucomannan porang will be fermented by microbiota in the intestine. Because of their ability to be used as a fermentation material for microbes, glucomannan is said to have probiotic activity, which is good for health. Seeing this potential and as a probiotic-based food ingredient, finally the idea emerged to make a diet treatment for people with overweight and obesity. The idea is, of course, that apart from being low in calories, because glucomannan has a very low calories, and also in the same time, it can also give you a satiety effect. For this reason, in this research, we will look at how the diversity of human gut microbial population is changed because of our intervention, and also we support our data with body weight loss. This is our method. The first is we will extract glucomannan from porang tuber into glucomannan flour, and then we will develop a jelly product that contains glucomannan porang itself, and then we will do clinical trial or food intervention on the people with overweight and obesity. This clinical trial will be done randomly, and we will divide it into two main groups. The first group is treatment group, treatment with jelly glucomannan porang, and the second group is placebo group. This clinical trial will be done in 8 weeks, preclinic week 0, and then mid at week 4, and the post week at the 8th week. We also do food recall every day and food intake monitoring and evaluation every 2 weeks. And also we will sampling the stool from the respondent and do anthropometry test every 4 weeks in week 0, 4th, and then 8th. We also have ethical clearance approval from Faculty of Medicine, Universitas Gadjah Mada, and Sarjito Hospital in Yogyakarta. This is the method for profiling microbiota, sampling phases in the 0, 4, and then 8th week, and then edit with RNA letter, storage at -20 degrees Celsius to prevent damage, and then DNA extraction using DNA isolation kit. And then we will check the DNA genome using gel agarose electrophoresis. After that, we will PCR using ribosomal intergenic spacer analysis using this primer, and also using this program of PCR itself. And then after that, we will get the DNA band, we will clustering it and make it into a dendrogram, and then we will choose the sample for sequencing using next generation sequencing Illumine Novasec from Singapore. The NGS that we use in this research is using 16S metagenome in the V4 region. And after that, after we NGS, we will analyze the data using KEYNOTE 2.0. This is the result that show the relative abundance from phyla level of the sample. This is there is a 6 sample in this slide, 3 from group A and 3 from group B. The group A is indicated the glucomannan porang jelly treatment, while the B indicated the placebo group. The results show that for all the sample, gut microbiota are dominated with phylum Bacteroidetes and Firmicutes. And after that, we will see at the group A first. In the group A, there is an increasing of Firmicutes after 4 weeks, and then decreasing again in the 8th week. While for the Bacteroidetes, there is a decreasing in the 4th week, but increasing in the 8th week. While in the B group or in placebo group, the result is different. In a B group, the relative abundance of Firmicutes relatively constant to decrease, while for Bacteroidetes, the relative abundance of Bacteroidetes in the group B is constantly increased, while for the other phyla in this result, there is no significant effect after intervention before or after intervention of glucomannan or placebo. In this slide, we will look at the gut diversity of microbes of the sample tested at the phyla and the genus level. The right picture shows the distribution of the heat map of phyla, while the image on the left shows the distribution of heat map at the genus level. In the left picture, we can see that jelly glucomannan porang group has higher abundance of bacteria than the placebo group in all almost phyla. This can be seen from the dominance of red color in group A and the dominance of blue color in group B. Apart from there, we can also see the intervention of jelly glucomannan porang given is able to reduce almost all type of phyla except for Bacteroidetes phylum. In Bacteroidetes phylum, as we can see that from group A, there is a decreasing after 4 weeks and then increasing again after 8 weeks, while in the B group of there is an constantly increasing of Bacteroidetes after intervention. The next result, while at the genus level shown in the image on the right, we can see that the distribution of number of genera of the two groups is different. This can be seen from the different distribution of blue and red between the two groups. Genera in blue in group A will have a red color in the group B and vice versa. Let's take a look at the Lactobacillus for one of the examples. Lactobacillus in group A is constantly increasing after the intervention of glucomannan porang, while in the placebo group, the relative abundance of Lactobacillus is decreasing from red to blue. So from this result, we can see that there is a different result between placebo and glucomannan porang group. There is a different result from sample with intervention glucomannan porang and sample without intervention of glucomannan porang. And this is the table of weight loss from the pre-test, mid-test, and then post-test. We can see the highest loss body weight is from group B, KG228 minus 6.7, while the lowest loss body weight is from group A with just minus 0.9. So we will see the correlation between the weight loss and profile good bacteria in the next slide. This slide shows a graph of the correlation between body weight and relative abundance of several dominant phyla. Let's say Firmicutes, Bacteroidetes, Actinobacteria, Proteobacteria, Verrucomycetobacteria, Chloroflexi, Acetobacteria, Cyanobacteria, and the other bacteria. Furthermore, the correlation between the correlation value can be seen on the next slide in accordance with Pearson correlation test shown in the next slide. This is the correlation Pearson test. This is the number of p-value between body weight and each phyla level. Body weight versus Firmicutes, body weight versus Bacteroidetes, body weight Actinobacteria, and so on. And this is the significant level, whether the result is significant or not, if there is correlation or not. From the Firmicutes, Bacteroidetes, and Actinobacteria, the correlation Pearson test resulting there is no significant correlation between body weight and these three phyla, while for the other phyla, the results show that there is a significant correlation because the number of p-value is quietly high. It's 0.9 blah 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 for all of this phyla, except Firmicutes, Bacteroidetes, and Actinobacteria. And the last, this is the picture of heat map. We want to make correlation between the time of intervention on each treatment, placebo and glucomannan porang treatment, to some of dominant phyla. in a human gut microbiota profile. From this result, we can see that in vermicutes, from the glucomannan porang treatment, the relative abundance of vermicutes after intervention is constantly decreased from the red to the white. And also for the placebo, the number or the relative abundance of vermicutes also decreasing, while in bacteroidetes, it's quietly different. In bacteroidetes, from the relative abundance of treatment group or glucomannan porang jelly group, there is a increasing of bacteroidetes, while in the placebo group also increasing of bacteroidetes. While in actinobacteria, proteobacteria is the same with vermicutes. But in Verrucobacteria, Chlorophaxi, Acidobacteria, Cyanobacteria, and the other bacteria, there is no significant difference before and after intervention. I think that's all of my presentation for today. Thank you for the attention. I'm sorry if I have many mistakes from this presentation. The last I say, thank you and see you next time.
Hello everybody, my name is Mahmoud Hollie. I'm here to present my abstract about coronavirus behavior in pregnancy and its management. In the beginning, I'd like to thank Mr. Dangso and Mr. Bashkangi for their supervision and their support to me during my abstract preparation. The COVID pandemic has affected the healthcare system globally and has got its implication on the mothers and their babies in pregnancy, and especially with the new evolving strains, besides the rarity of the available data. The aim of this abstract is to share our experience and learning points from our case report in Scunthorpe General Hospital. This is a case presentation of a 25-year-old lady, 33 weeks pregnant, who was admitted with the severe symptoms of adult respiratory distress syndrome 5 days after being tested positive with COVID-19. Given the severity of the ARDS she had, an emergency C-section was planned. However, with prompt planning and MDT cooperation involving 9 different clinical teams, her symptoms improved and the delivery was postponed. 9 days later, she was discharged home and she delivered electively at term. This is a table shows 7 case reports of severe COVID-19 in pregnancy. 3 of those cases have delivered vaginally, and 2 of them required to be on ventilators antenatally. One of them required it for 23 days and the other one required it for 10 days. But they still had a complete recovery afterwards. The other 4 cases had emergency C-section. One of them showed postnatal deterioration and she required extracorporeal membranous oxygenation because of failure of a mechanical ventilation. And another case had a complication on the baby who died 5 days postnatally with septic shock and respiratory failure. These evidences come in accordance with our conservative approach and suggest that iatrogenic delivery can be avoided. Also, the severity of the presented complaints are not always necessity for an obstetric intervention as proved. In conclusion, the viral behavior is not completely understood so far, and we're waiting for new researches and case reports. But evidences show that NDT meeting and constant care are the cornerstone for successful management of every challenging COVID-19 pneumonia in pregnancy. Thank you.
Hi, my name's Laura, and I'm going to be talking about my research where I was testing for respiratory viruses other than SARS-CoV-2 in symptomatic healthcare workers. Since the beginning of the pandemic, healthcare workers with symptoms of a cough or a cold have been screened for SARS-CoV-2. This is the first time we've screened staff members for respiratory infection before allowing them to return to work. This got me thinking, why should we only test for SARS-CoV-2? There are plenty of other respiratory viruses which are highly infectious, spread easily through hospitals, and cause life-threatening infection in our vulnerable patients. I wanted to find out whether screening symptomatic healthcare workers using a full respiratory virus PCR panel would provide any useful additional data for infection control. I tested 333 stored occupational health samples, all of which were residual MPS fluid, or nasopharyngeal swab. All were symptomatic healthcare workers who attended the SARS-CoV-2 staff testing clinic at Gosh between September and November 2020. All were screened for SARS-CoV-2 as part of their routine service, and 10 of them tested positive. I didn't do any further SARS-CoV-2 testing, but I did test for RSV A and B, human metapneumovirus, influenza A and B, parainfluenza 1, 2, and 3, adenovirus, rhinovirus, and enterovirus. And these are my results. So as we can see, 194 out of 333 healthcare workers tested positive for a respiratory virus other than SARS-CoV-2. This is nearly 60% of those tested. The majority of these tested positive for rhinovirus, and most of these infections occurred during September, which is also the time when children returned to school after 6 months of social distancing and homeschooling. I also identified 7 co-infections, all of which were adenovirus plus rhinovirus. Interestingly, none of the healthcare workers which tested positive for SARS-CoV-2 tested positive for any other respiratory virus. So to conclude, my research has shown that screening healthcare workers for SARS-CoV-2 alone will miss other highly infectious respiratory viruses. 194 out of my 333 healthcare workers would have been sent back to work with patients and colleagues whilst positive for another virus. This may not have been appropriate or safe. Screening using a full respiratory virus panel would yield clinically actionable results which could be utilised by infection control. Now, infection control are already incredibly busy, so we could consider prioritising this testing during the winter months when most of these viruses circulate, or we could prioritise testing in healthcare workers who work with the most vulnerable patients, for whom these viruses would be the most dangerous. The control measures taken against SARS-CoV-2 in September 2020 were not effective in controlling the spread of rhinovirus. We saw a big increase in the number of tests being requested for SARS-CoV-2, but we didn't see an increase in positivity rates. My research shows that this is probably because of high rates of rhinovirus in the population instead. Viral co-infections with SARS-CoV-2 are rare in otherwise healthy individuals. This suggests that if you test positive for one of the other respiratory viruses, you are unlikely to be infected with SARS-CoV-2. Again, this is useful for infection control. Thank you for listening, and my email address is here if anyone wants to get in touch.
Hi, this is Mohammed Al-Khalidi from the UAE University, and we'll talk briefly about E. coli 0157H7 phage from bird feces and its potential application as a therapeutic agent and as an industrial food preservative. E. coli 0157H7 is a famous foodborne pathogen which was responsible for many outbreaks worldwide recently, causing severe hemorrhagic colitis leading to hospitalization, yet no antibiotics are recommended because this bacterium is full of toxin and you don't need to release these toxins to the body to worsen the case. Antidiarrheal agents are not recommended either. So what is the solution? The best solution is to eliminate this bacterium during the food process. The use of bacteriophages as food biopreservatives is well established, and many phage products as biopreservatives have been approved by the US FDA and commercially available. Therefore, scientists all over the world are scanning ruminants' menus for a specific E. coli 0157H7 phage. We changed the game. We shifted from ruminants to birds, and guess what? We found 3 bacteriophages that target E. coli 0157H7. Here we are going to introduce the most specific one, named UAE MI01. Please have a look. at the background pictures. These are TEM pictures of this phage. This is the first time in the history to report E. coli 157H7 phage from a bird. So, why this phage can be an ideal biopreservative? The whole genome sequencing of UAE MI01 phage revealed that its genome consists of 44,281 base pairs with an average GC content of 54.7%. So this is a very small bacteriophage. With an eclipse period of 10 minutes, a latent period of 50 minutes, with birth size of about 100 plaque-forming units. The phage is stable in various challenging situations, for example, in temperatures ranging from -20 to 60 Celsius and in pH levels from 4 to 10, which make it an excellent biopreservative in food process. Please do not hesitate to contact us if you have any question or comments. Thank you.
Good day all. I'm Dr. Nuhu from Usman Danfodio University, Sokoto, Nigeria. My topic is on assessment of thrombocytopenia and anemia in HIV-positive patients on highly active antiretroviral therapy. Human immunodeficiency virus, HIV, is a retroviral. HIV-infected person becomes an easy target for opportunistic infection and diseases. HIV and acquired immune deficiency syndromes, AIDS, remain the most challenging significant infectious agent in the last century. Hematological complications have been documented to be the second most common cause of morbidity and mortality in HIV patients, and are generally marked with cytopenia such as anemia, neutropenia, lymphopenia, and thrombocytopenia. Drugs have been documented to have cytotoxic effect, especially when used as immunotherapy. However, the study is aimed at assessing thrombocytopenia and anemia on HIV-positive patients that are placed on highly antiretroviral therapy attending General Hospital. The study is hospital-based cross-sectional study among HIV patients. The study was concluded within 3 months, January to March 2020. Laboratory methods include HIV detection using serial algorithm according to World Health Organization and the full blood count analysis using Sexmed SP300 analyzer. The result is indicated here in the table. The mean standard deviation of hematological parameter of the subject in order of establishing thrombocytopenia and anemia indicated that HIV-positive patients are prone to thrombocytopenia and anemia, except for being HIV-positive or negative does not affect the neutrophil. There is no significant hematological parameter, CD4 count, and viral load that is subject to age. CD4 count and viral load are independent. Despite the usage of HAART by HIV-infected patients, they are still prone to thrombocytopenia and anemia. However, the viral load, CD4 count are independent of the age and sex. In conclusion, thrombocytopenia and anemia seem to be a common feature in HIV-positive patients, as it was observed in this study, that can lead to bleeding and difficult therapy for other pathologies. Diagnostic screening for factors related to the virus co-infection for genetic diseases are still needed for this specific population. Thank you all.
I am Dr. Bola from Department of Pharmaceutical Microbiology, University of Ibadan. My topic is anti-measles activity of 4-hydroxy-3-methoxybenzaldehyde isolated from Xylopia ethiopica. This is the structure of measles virus. This is the picture of the plant Xylopia ethiopica. Justification. Measles is a communicable disease caused by measles virus. Although clinically effective live vaccines are currently in use, measles still remains a major cause of morbidity and mortality in children below 5 years of age, with death rate of 4% worldwide. Approximately 145,700 children under 5 years die from measles in 2013. Recently, over 1,158 children were reportedly infected with measles in northern Nigeria. Oluremi and Andrade-Linares in 2015 reported that hexane and dichlorometane fractions of Xylopia ethiopica leaves showed significant activity against measles virus with IC50 values of 1.246 and 0.691 microgram mil. Methods and results. Antiviral assay of active fractions was carried out on viral cells by tissue culture techniques using inhibition of cytopathic effect model. Ability to inhibit viral-induced cytopathic effect in tissue culture was evaluated 72-hour post-infection colorimetrically using MTT dye, and optical density values were determined using multi-well spectrophotometer. Compounds in bioactive fractions were isolated using thin-layer and column chromatography and characterized spectroscopically. 4-hydroxy-3-methoxybenzene carbondaldehyde, vanillin, was isolated from the dichlorometane fraction of Xylopia ethiopica leaf. Vanillin was active on measles virus with IC50 of 0.71 microgram per ml and selective index of 118.56. It was found to interfere with viral attachment and fusion. Impact on the society. 4-hydroxy-3-methoxybenzene carbondaldehyde would be a promising antiviral lead compound in developing anti-measles agents to manage accidental exposures and periodic outbreaks of measles. It will significantly contribute to reducing the burden of measles in Nigeria and globally. It might be useful as a potential template for developing novel antiviral agents. Thank you for listening.
Hello, my name is Pawni. I am from Sri Lanka. First of all, I want to thank you all for giving me this wonderful opportunity to present my research abstract in this esteemed research conference. So now let's move on to my abstract. The title is An Escalation of Hantavirus Disease Burden Among the Leptospirosis Suspected Patients in a Tertiary Care Hospital Setting in Sri Lanka. So, what is hantavirus? Hantavirus is a rodent-borne acute febrile infection, and hantavirus infection is distributed all over the world. And hantavirus has an important fact, that is the virus has the capability of co-evolving with its host. So, as example, the deer mouse has the cynomrovirus. Bank walls has the Pumala virus, where the yellow-necked mouse had the Dobrova virus. So if we move on to the disease transmission, as you can see in this image, when the rodent drop their feces in outer environment, when it get dried up, the virus particles get mixed up with the air. And when we inhale these contaminated air particles, we get the virus. This mode of transmission is known as airborne transmission. Apart from this, you might get the virus when you are bitten by an infected rodent or when you touch the surface infected with the rodent who has the hantavirus infection. But these type of infections are very rare. The main method of transmission of a hantavirus is airborne transmission. That is why hantavirus infection is more threatening. So if we talk about the clinical manifestation of the virus, hantavirus has two clinical manifestations. Hemorrhagic fever with renal syndrome, which is commonly known as HFRS. HFRS is caused by a group of hantaviruses known as all world hantaviruses. And this type of hantavirus infection is mainly distributed in European and Asian regions. And other type is hantavirus pulmonary syndrome, which is simply known as HPS. And HPS is caused by New World hantaviruses, and it is prevalent in America, North and Latin American region. And if you got HPS, your lung will be attacked by the virus. And if you get HFRS, your kidneys will be damaged. So HFRS is mainly caused by 4 types of hantaviruses: Hantan virus, Dobrava Belgrade virus, Humala virus, and Sol virus. And Synombra virus is the cause of most of the HPS cases in America. And there is a milder form of HFRS prevalent in European region. It is known as Nephropathica epidemica, and it is caused by the Humala virus. So if you move on to the clinical phase of HFRS, HFRS has 5 to 10 days of incubation period, and it has 5 phases: incubation, febrile phase, hypertensive phase, oliguric phase, polyuric, and convalescent. And if we talk about the hantavirus infection symptoms, you get fever, you might get headache or myalgia, diarrhea or backache, any kind of pains, like common cold and symptoms like symptoms we are getting when we have a leptospirosis infection. That is in the febrile phase. After that, you are getting into shocks and hemorrhages or renal failure. Since I'm talking about the hantavirus infection in Sri Lanka, I think it's best if I share more details of hantavirus infection in Sri Lanka. This is the map of Sri Lanka, a small island in Indian Ocean. The first reference of hantavirus infection is in 1988, where in Colombo they have found the evidence of Sol virus in few rats collected. And after that, in 2011, there was a serological study where they have found Thailand-like hantavirus is circulating among the central cities around in our small island. And in 2014, there were evidence of Humala virus infecting the febrile patients in northern part of the country. And in 2017, there was an important study done by Dr. Magee, where there was a hypothesis that hantavirus infection could progress into the chronic kidney disease with unknown etiology. And they have found some interesting results that more than 50% of CKDU patients had hantavirus infection. So from these results, we can say that Sri Lanka did not have many studies in regarding hantavirus. Here, some few studies, but all of these studies found few evidence of prevalence of hantavirus infection. So there's one study I want to point out. This was conducted in same tertiary care hospital setting where I found my research findings, and this study was conducted by my supervisor when he was a postgraduate student. And here in 2019 in Kandy, a teaching hospital, there he found among 105 leptospirosis suspected patients, 8 had hantavirus antibodies. As a percentage, it's 7.6%. So now let's move on to my research. Duration of the study was 2015 to 2020, and the hospital setting was same setting I mentioned earlier, a teaching hospital in Peradeniya, Kandy, and sample type was human serum, and the test where I have used was indirect immunofluorescence assay, and the cells I used as antigen was Thailand ortho hantavirus infected Vero E6 cells. So we will be able to get the details whether these people are infected with Thailand-like hantavirus. So here you can see an image of how a positive sample, how a hunter positive patient sample look under fluorescence microscope and how a hantavirus negative sample look under fluorescence microscope. And as I already mentioned, hantavirus clinical symptoms are almost similar to leptospirosis. So in order to confirm an infection, a serological test is mandatory. So the method is rather simple. We selected patients with leptospirosis symptoms, and we conducted ELISA to find whether these people have leptospirosis infection, and we remove patients who have leptospirosis infection from our study group, and we only selected people who are not having leptospirosis, and then we did IFA to them and calculate and found out the number of people who are infected with hantavirus, and we calculate this amount for 5 consecutive years. So when we move on to the results section, you can see apart from 2015, all other 5 years had a more than infection of more than 20%. So if we take a rate or the more common value, the rate of the infection, the mean value is more than 20%. It's nearly 20.8% for these years. And in 2020, the number of total patient number is very low because of the COVID-19. So finally, I want to tell you that from these results, you can also see that hantavirus infection is mostly misdiagnosed, or it is an underreported zoonotic disease. So if we compare the result from the 2019 study and the result from this 2015 to 2020, you can see that this disease rate is escalated drastically. And the most important thing is we do not have enough diagnostic facilities to find out the infection rate of hantavirus. And mostly because we don't have any financial means of founding these things. Only 1 or 2 places in whole country is capable of conducting IF. And one thing we are capable of doing is educating the general public on prevention methods. So I want to acknowledge my supervisor, Dr. Chandika Gamagiri, for expert supervision, and also Professor Kumiko Yoshimatsu for giving these valuable materials for this research, and of course the technical officer who conducted this IFS studies. And these are the references I use for this research. And finally, I want to thank you all for listening to my research presentation. And of course, if you have any question regarding hantavirus infection, please send an email to my email address. I will try to answer your questions by best of my knowledge. Thank you all.
Hello everyone, I am here to present you the results of our study recently published at IGFM, where we have evaluated antimicrobial resistance determinant and virulence factors in 37 Salmonella enteric strains involved in outbreaks in Brazil using whole genome sequencing. The phylogenetic analyses were performed for those sorvars recognized as prevalent in outbreaks in the country using publicly available strains, isolated in Brazil or in other countries. The resistance analysis revealed resistant genes to a range of antibiotics, including fosfomycin, tetracyclines, and also the plasmidome showed a range of different plasmids, and we identified 8 Salmonella pathogenesis islands and more than 100 genes related to virulence or stress response in the genomes evaluated. Here we report for the first time the K88 gene in some of the serovars evaluated and the first report of PLW in S. pulmona. In the phylogenetic analysis of Salmonella infantis, we observed two clusters. Our strains were grouping in the first cluster, the cluster A, with other strains isolated later in the country, suggesting a 40 years relationship at the environmental food-human interface for these strains in Brazil. For the S. Johannesburg, we observed that our samples grouped with other samples from US, UK, and China, isolated from a range of different sources, suggesting an intercontinental dissemination since at least 2002. And finally, for Salmonella typhimurium, we observed a higher diversity, and our strains grouped in 3 different clusters with strains isolated in the country from poultry samples, and suggested that these strains were circulating in the country for more than 30 years, grouping with strains isolated, for example, since 1985. Therefore, our results extend the current knowledge regarding the Salmonella enteric strains, antimicrobial resistance determinants. We report for the first time specific genes in Salmonella enterica serovars, and our findings highlighted the virulence and the antimicrobial resistance determinant that may lead to recurring food outbreaks in the country.
Hello everyone, I'm Yemisi Jeffagbola from the Department of Biological Sciences, University of Medical Science, Ondo. I'm here to talk about the position of African women in mycotoxin research at International Applied Microbiology Conference. And we know that ensuring food safety is a public health priority and is essential steps to achieving food security, and this can help us to safeguard the health and well-being of people, fostering economic development and improving livelihood. And the aim of this presentation focuses on highlighting the role played by women of different categories to ascertain food safety and mycotoxin prevention as one of the tools towards increasing food sufficiency and curbing crisis in Africa. Why do we focus on women in Africa? We focus on women in Africa for mycotoxin control because women process about 60 to 80% of food, and they work in farms and other agricultural activities. They process foods, they prepare foods at home, and this has to be like women play an essential role in prevention of foodborne disease in Africa. And what is mycotoxin? Mycotoxins secondary metabolites. I call them poison. They are toxic secondary metabolites produced by toxigenic fungi of toxigenic microorganisms like toxigenic fungi, and they are capable of causing disease and death in both humans and animals. There are different types of mycotoxin. We have aflatoxin, caused by Aspergillus flavus, Aspergillus parasiticus, Fumosin, T2 toxin, and several mycotoxins. And the role of women in mycotoxin management in Africa include like the timing of planting, the help in post-harvest, drying, storage, processing, fermentation, packaging, and income. And so therefore, African women are the custodians of traditional knowledge in terms of food preservation, and since women do all the basic needs of the households, and so they are the ones that are responsible for mycotoxin prevention in food. And several interventions has been carried out in the research we have done in order to prevent mycotoxin occurrence. Like we have research about most of the women we have researched about better drying method in controlling toxigenic mold contamination, and we've been able to develop biocontrol formulation from cassava peels formulated into pellets, and this will serve as for feed control of Aspergillus flavus And in conclusion, we discover that awareness creation, education, and capacity building will assist women scientists and farmers in carrying out some kind of research work that will prevent mycotoxin contamination. Thank you for listening.
Hello everyone, my name is Urszula Zarzecka and I'm PhD student from Chair of Industrial and Food Microbiology at the University of Warmia and Mazur in Poland. I would like to present my research conducted together with my supervisions about the antibiotic resistance in bacteria from starter cultures and their ability to resistance genes transfer. Many starter culture strains are considered as traditional food ingredients due to the long history of safe use, so they are approved for use in the food production within the European Union with no need to carry out studies confirming their safety. However, there is no information about the occurrence of antibiotic resistance genes in these strains. So the objective of the study was to determine the antibiotic resistance pattern of the strains from the commercial starter cultures, analysis of the antibiotic resistance genes occurrence, and to investigate the ability of horizontal transfer of the resistance genes. According to the research hypothesis, strains used in the food industry as starter cultures may be a reservoir of antibiotic resistance genes and participate in their horizontal spread. The research material consisted of 44 strains from 30 starter cultures. The majority of the strains belong to the Lactobacillus and Lactococcus genus. The study consisted of 3 stages. In the first one, the minimum inhibitory concentrations for selected antibiotics were determined using gradient strips and the Vitek2 compact system. Then the frequency of the resistance genes was determined using PCR. In the last step, the possibility of the resistance genes transfer in vitro and in situ was analyzed. The following results were obtained. The highest MAIC values were observed for the tetracycline, kanamycin, and chloramphenicol. Analysis of the resistance genes occurrence showed that more than half of the strains carried the gene encoding resistance to kanamycin and to chloramphenicol. A large percentage of the strains also had the gene encoding resistance to gentamicin and tetracycline. The greatest diversity was observed in the occurrence of tetracycline resistance genes, as 6 different TET genes were found in the tested strains. For the analysis of the horizontal gene transfer possibility, 3 strains were selected as donors. They carried the TET-K and TET-M genes with plasmid localization. It was found that the gene transfer took place both in vitro and in situ, but in vitro transfer had larger frequency rate. In conclusion, antibiotic resistance may occur among the starter culture's microorganisms. Ability to resistance genes transfer was also demonstrated, which may contribute to the spreading of the resistance in the environment. This indicates the need for further research in this area to be sure that the strains introduced into food do not constitute an environmental reservoir of antibiotic resistance genes. Thank you very much for your attention. If you have any question for me, please feel free to contact me.
Hi, I'm Sa Choo, and this presentation is on an introduction of carbapenemase producing organisms external quality assessment scheme by UK NEQuest for Microbiology. And this research was done with Jennifer Henderson, Sheila Seaton, and Mohammed Islam. So why do we want to create a carbapenemase producing organisms, or CPO for short, scheme? So the carbapenems are broad spectrum antimicrobials that are frequently used as a last resort treatment for life-threatening nosocomial infections. As they're often the last effective defense against infections caused by multi-drug resistant bacteria, their increased and often inappropriate usage has led to the development of resistance through acquisition of carbapenemase enzymes. This increased CPO prevalence has led to a need for diagnostic labs to perform local detection of the major carbapenemase families, hence the need for an EQA scheme to assess the performance of these labs. This preliminary research has mainly focused on validating the use of swabs to distribute specimens. In a clinical setting, screening patients for CPO would typically involve testing a stool sample or rectal swab. This new scheme therefore endeavours to distribute specimens as swab types, this being a more authentic or representative sample type. So we sent out a pre-pilot survey to laboratories to assess interest in a pilot scheme. Of 394 responses received, 327 expressed interest in participating. We then set about carrying out stability testing on some different swab types. The swabs we looked at were a liquid AMI swab, dry swabs in brain heart infusion broth, and fecal trans swabs. So we wanted to look at whether any of these swabs were a feasible form for us to send specimens to participating labs. We prepared 6 selected CPOs in both a lyophilized freeze-dried form and with each of the swab types listed previously. So these were stored at 4 degrees Celsius and at room temperature, which was between 18 to 25 degrees Celsius. At set time points for 8 weeks, we assessed the viability of the organisms by placing them onto McConkey agar and also onto selective chromogenic plates. We noted the morphology and the growth after incubation at 37 degrees Celsius and used the rapid NG Biotech Carbo5 test to detect carbophenamases. The results indicated to us that the liquid AMI swab stored at room temperature was most comparable to that of the freeze-dried specimen. It showed good viability throughout the 8 weeks, so we can now move forward with launching the pilot scheme using both a freeze-dried specimen and a swab sample, and this will further enable us to determine the suitability of the swab sample for the eventual launch of the scheme.
Hi, I'm Muralidharan Velappan. I'm a microbiologist. Presently, I'm a research scholar at University Chennai. Here I am going to present a 3 minutes flash presentation on toxicological interactions of micro and nanoparticles of freshwater fishes, current status of knowledge with the focus of ecological relevance. Globally, the presence of micro and nanoplastics are frequently detected in aquatic organisms. Despite well studied among marine fishes, their roles and their significance in freshwater fishes remains unclear. To understand the toxicological interactions of microplastics and nanoplastics in freshwater fishes, so which affects their physiology and behavior, further this will introduce them into food chain. About 94% of all freshwater fishes occurs in developing countries, providing food chain and livelihood for millions of the world's poorest people and contributing to the overall economic and well-being by means of exploration, tourism and recreation. Experiments have demonstrated some degree of toxicological and pathological different effect observed in freshwater fishes. Behavioral abnormalities in terms of feeding and movement of adults and larvae, as well as the reproduction in adults. There are evidence mother and offspring transfer of nanoplastics and the parental exposure of microplastics affected early developmental of the neonates. The histopathological evidence shows some micro and nanoplastics were accumulated in terms of the adult gut and in some cases in liver and gill. So micro and nanoplastics could be the internalized and the cause of detectable biomarkers changes in the blood cells. In rare cases, micro and nanoplastics have also found in fish's brain and caused changes in the brain appearance showed significantly inhibited acetylcholine esterase activity. There is ample evidence of concentration dependent acute toxicity as well as the chronic effects. Further plan and collaborations attempts between ecologists and epidemiologists to study the bioaccumulations of a micro and nanoplastics in human via the food chain is various geographical location would be also recommended.
Hello, this presentation is part of the online International Applied Microbiology Conference, which takes part from the 7th to the 11th of June. My name is Lidia Paleodimou. I'm an MSc Microbiology graduate from Northumbria University, and in today's presentation we will talk about our published research in the field of Listeria environmental persistence, and more specifically in the link between the genetic markers to the persistence phenotype and to the strain virulence potential. Our presentation is comprised of a short introduction, the most significant results and discussion of our work, and the conclusions. To begin with, Listeria species are divided into two categories, the pathogens and the non-pathogens. Listeria monocytogenes and Listeria ivanovi are two pathogenic Listeria species which can cause disease in humans and ruminants, or ruminants only, although some rare Listeria ivanovi human cases have been recorded in the past. The non-pathogenic species are comprised of 19 other Listeria species, including the environmental Listeria walsmeri and Listeria inokia species. However, one of the highest significance is Listeria monocytogenes, which is the causative agent of listeriosis. Listeria monocytogenes is a gram-positive and rod-shaped bacterium, as you can see in the picture, and it was first isolated in 1926 during an outbreak in animals, while in the 1970s it was identified as being responsible for the clinical cases of foodborne diseases, and in the 1980s it was classified as being a foodborne pathogen. So Listeria monocytogenes is a facultative anaerobe, and it can grow in various environmental conditions, like in both high and low temperatures and pH values, in high salt concentrations, and all of them pose it as resistant to various environmental stresses. Listeriosis is a foodborne intracellular and rare disease with a very high mortality rate that is 20 to 30%, meaning that 1 out of 5 people typically dies from this invasive disease. It causes a variety of symptoms, including mild, like gastroenteritis, to more severe ones, like meningitis or septicemia, and it is usually the risk groups who are more susceptible, including the elderly, the immunocompromised, and the pregnant women with their fetuses, since they can be contaminated via transplacental infection. So, persistence. As persistent are defined the strains which show an ongoing environmental contamination in the food processing environments, despite the sanitization measures. So, in a food processing environment, like the one you can see in the picture, there can be two contamination types. The first one are the persisters. The persisters are those strains who have been isolated multiple times from the facility over different time points. And the second one are the non-persisters, which have been isolated only once from the environment, usually over long periods of surveillance, for example years, like in the case of our study. So, when a persistent isolate is both hypervirulent and pathogenic, the possibility of an outbreak is increased, and this higher risk is due to the repeated dissemination of the persistent virulent strain throughout the food processing environment. So, the strain has a greater chance of contaminating the food products and causing an infection or an outbreak of the disease. But what are the factors leading to the persistence? Some of the most possible explanations are the poor hygiene, the formation of biofilms, and the presence of genetic markers promoting resistance to environmental conditions, like, for example, disinfection, which may be carried by mobile genetic elements, like phases or transposons, for example. So, the first and one of the most important steps to diminish the chances of persistence is an effective hygiene regime. Therefore, the food facility is first cleaned well and then sanitized. The isolates in our study have all been taken from the same environment, which underwent sanitization based on a rotation procedure comprised of acid, alkaline, and quaternary ammonium compound-based disinfectants. So, we interrogated the draft genomes of 13 Listeria species comprised of Listeria monocytogenes, Listeria inokia, and Listeria walsmeri, which were both persistent and non-persistent, through bioinformatic analysis. And as was mentioned previously, all of them were isolated over a 2-year period of surveillance from a poultry food processing environment. So all of the strains were under the same selective processes. And our aim was to identify whether the presence of genetic markers linked to stress resistance, for example, disinfectant resistance, or for markers being carried on mobile genetic elements, have an association to the persistence phenotype in the Listeria species. So, in the following slides, the most important of our findings will be presented. The figure 1 is a heat map presenting the data of the presence and absence of a number of genetic markers related to some of the key stressors that we looked in our study. These are, for example, the disinfectants, antibiotic resistance, heavy metal resistance, as well as temperature conditions. So, as you can see from this figure, genetic markers were widespread in the population without any specific correlation to persistence. Regarding disinfection, we can observe that different disinfectant genetic markers are prevalent in the population, but what is of interest in this table is the Quack-8 gene. The Quack-8 gene is a disinfectant stressor expressing resistance to quaternary ammonium compounds, which are compounds found in one out of the three categories of sanitizers used for the cleaning of the food facility. So, previous research has shown that the Quack-8 gene is carried on the transposon TN6188, in which we will refer more in a while. The heavy metal resistance is another important feature of Listeria in the food systems due to their presence in the feed and the fertilizers, for example, and they may be disseminated through the population via mobile genetic elements. However, there has not been a direct link or correlation between cadmium or arsenic resistance and persistence, and our findings do not suggest an association too. But what is of interest is that the CAD-1 gene is more common on persistence, whereas the CAD-4, which is known for providing lower tolerance, was only found in the non-persisters. Regarding arsenic, the presence of the transposon 554, which carries the arsenic resistance cassette in persistent isolates, could possibly be due to horizontal gene transfer events. So, regarding the COMK gene now, the COMK gene is a known phase insertion hotspot in the Listeria monocytogenes species, and the COMK phase has been suggested to have a role in persistence by functioning as a rapid adaptation island. However, the COMK phase in our study was found almost equally distributed among persisters and the non-persistent strains, suggesting that there is no clear link to persistence. However, I'd like to draw your attention to a particular point of interest. As you can see in this figure, the Listeria Waltzieri isolates had no COMK phase insert, and after further comparative and genomic analysis, we found that Listeria Waltzieri isolates have a mutation in their ATTB site. So, in order to investigate if this single nucleotide polymorphism was conserved across other strains of Listeria waltzieri, and to determine if a COMK insertion was present in other Listeria waltzieri strains, we examined genomes available in the NCBI database. As you can see from this analysis, all strains harbored the same SNP and none had COMK mutations. So, this may indicate that this common Listeria species phase insertion is rare or totally absent in Listeria waltzieri strains. So now let's go back to the transposon. The transposon TN6188 has been associated with persistent strains in the food processing environments, and in our research it was harbored by the persistent Listeria monocytogenes UCDL019, which is an ST121 isolate, and the Listeria inokia UCDL146. It has been suggested that the presence of disinfectant-resistant markers is not the sole causative mechanism of persistence, but actually what is of interest in this figure is that two different Listeria species share a transposon 6188 with 100% similarity, while a previous study examining the distribution of this genetic element among Listeria species only identified it in Listeria monocytogenes. So, our study demonstrated that this mobile genetic element can be harbored by other Listeria species, in this case Listeria inokia, indicating that this transposon is not solely found in Listeria monocytogenes, but other Listeria species could act as reservoirs for such genetic markers, possibly linked to persistence. This emphasizes the need to monitor for and control all Listeria species, as even non-pathogenic strains can pose a food safety risk for listeriosis through acting as environmental reservoirs for stress-resistant related genetic elements. So, another valuable part of our study are the stress survival islets. In figure 5, you can see representation of the SSIs which were present among our isolates. In general, the SSIs have been previously suggested as important for the persistence of Listeria in the food facilities, and in particular due to their benefits related to survival under a variety of stress conditions like low pH, which is supported by the SSI-1, alkaline pH covered by the SSI-2, and oxidative conditions which are covered by both the SSIs. So the stress survival islets were prevalent across the Listeria population, which would also explain why some isolates persist despite the sanitization procedures. So what is of interest here too is the presence of a novel SSI in the non-persistent Listeria monocytogenes UCDL037. However, further experiments are required to identify if this plays a role in their environmental fitness or not. And finally, another very interesting question is what happens when persistence and virulence are combined. So, in figure 6, some of the virulence markers interrogated in our analysis are shown. The isolates on the top of the line are the persistence, while these on the bottom of the line are the non-persistence. And as you can see, there is an asterisk in some genes among the Listeria monocytogenes strains, and this actually indicates mutations in the genes of the ACT-A, internal A, and PRFA. These genes are involved in invasive disease, and strains with mutations in these genes are typically hypovirulent. Therefore, what we observed is that all the persistent isolates of the human pathogen Listeria monocytogenes are hypovirulent, suggesting that these strains have reduced virulence potential, and so the risk to public health is lower. However, this poses the question if strains of Listeria have diverged to evolve improved environmental fitness at the expense of pathogenicity, which needs more research. So, all in all, what we concluded is that there is not a single genetic marker observed that could promote persistence. The presence of coag-8s in the transposon TN6188 in Listeria monocytogenes isolate is not enough, and more research is needed to identify exactly how persistence is achieved. However, the finding of the transposon 6188 in a Listeria inocuo isolate supports the need to include more Listeria species in Listeria food safety programs. And similarly, the SSIs, the presence of the SSIs is a valuable part of our study and provides some useful insights in terms of persistence, but again, more research could validate better their significance. And finally, what can be derived from our research is that the persistent Listeria monocytogenes isolates have acquired traits and mutations suggesting hypovirulence and therefore less hazard for the human health. Nevertheless, Listeria persistence is a field requiring more in-depth research in order to identify exactly how and why Listeria species manage to survive and replicate in the food processing environments. So, this is the end of the presentation. Thank you very much for your time, and I hope you enjoyed and found it interesting. Please contact us if you have any question. Here are our contact details. Thank you very much and have a nice day. Goodbye.
Hello, my name is Richard Balsramanian and I am an MPhil student in veterinary sciences studying at the University of Cambridge. I'm excited to be sharing my presentation with you today. But before I get into my topic, it's important to discuss what antimicrobial resistance actually is. So AMR is usually caused by the overuse of antibiotics that introduces a selective pressure allowing for drug-resistant pathogens to survive and propagate. In humans, this usually manifests as the overprescription of drugs to treat illness, but in animals, antibiotics can be abused as growth promoters. While the United Kingdom has banned the use of antibiotics as a growth promoter, they are still critical for animal health. Therefore, it's actually vital to understand the effects of clinical uses of antibiotics on the spread of AMR to ensure that antibiotics can still be used as effective therapeutics. Specifically, we are interested in understanding how AMR mobilizes between microbial communities as a result of treatment. So the microbiome of livestock actually acts as an excellent platform to investigate this question because it is a known repository for AMR genes. As such, conventional metagenomic methods have been used to mine for AMR genes in the gut microbiome by simply isolating bacteria from animal byproducts, extracting genetic information, and then sequencing it, as we can see here. However, most AMR genes are actually located on mobile genetic elements like plasmids, seen here in red, and so through conventional methods, we actually have no way of associating a particular plasmid to its bacterial host species. Being able to identify where a particular AMR gene originated from is critical in order to allow us to ask dynamic questions about how these genes spread between bacterial communities through time. So Hi-C sequencing actually allows us to neatly circumvent this issue through a critical cross-linking step. Hi-C is a chromosome conformation capture technology that used to be used exclusively in a eukaryotic context, but here we've actually leveraged this in a prokaryotic context to associate genetic information that is in close proximity to one another. Through this critical cross-linking step that we can see here through the use of formaldehyde. So by cross-linking plasmids to their host DNA and then through sequencing of these hybrid genetic structures, we can actually identify where a mobile AMR gene was originally located. And so this actually acts as the basis for our study. So in our study, we collect fecal samples at multiple time points prior to and following antibiotic treatment in 6 BQP farms across the United Kingdom, and then generate Hi-C datasets for each time point that we've collected fecal samples for. So doing so actually allows us to not only understand how AMR gene frequency and bacterial species abundance changes through time, as could have been accomplished through any other generic metagenomic study, but we are actually critically able to understand how AMR genes spread between bacterial species through time, and this information can then act as the basis for a model of horizontal resistance gene transfer, which makes this work particularly novel. So thank you so much for your time. If you have any more questions, please feel free to contact me at the information I provided. Thank you.
Greetings, ladies and gentlemen. I'm Busu Swandaba from the Agricultural Research Council in Pretoria, South Africa. The topic for my today's presentation is on the interaction between phytosynthesized nanoparticles and rhizospheric microbes as plant growth promoters. The challenge of the world's population increase exacerbate climate change, increasing soil disturbance, thereby leading to soil nutrient deterioration, causing hunger and malnutrition. Hence, we need to find sustainable agricultural practices through restoring our soils for increased crop production. So nanostructured materials have been shown to play important roles in plants' physiological processes such as plant growth. However, it still remains elusive as to how these nanomaterials affect beneficial microbes in the soil. Hence, the study looks at the effect of phytosynthesized magnesium and iron nanoparticles on selected plant growth promoting microbes. We investigated the use of Artemisia plant extract for its efficacy in synthesizing nanoparticles. The extract was mixed with either iron or magnesium salt solution. The solution was then dried and annealed to form crystalline nanoparticles. Then these nanoparticles were tested for their efficacy on plant growth promoting microorganisms. Then nanoparticles were further investigated for their characteristics using TEM and EDS. TEM results in figure 1 showed monodispersed nanoparticles. EDS analysis in figure 2 confirmed iron and magnesium oxide elements. Nano-efficacy test confirmed that lower concentrations of both nanoparticles did not have an impact on the growth of selected microbes. However, increased concentrations of iron inhibited the growth of bacillus. In conclusion, quality nanoparticles were produced through phytosynthesis. More characterization techniques will still be conducted on the tested nanoparticles. More beneficial microbes will still be tested against their prepared nanoparticles. Overall, the study proved that toxicity of nanoparticles is concentration dependent. Ultimately, these nanoparticles will be tested as nanofertilizers for improved plant growth in greenhouse trials in concentrations that are not harmful to the tested microbes. We would like to thank the L'Oreal UNESCO for Women in Science Foundation, as well as the National Research Foundation South Africa for funding this research. Thank you.
The Good, the Bad and the Ugly. Antibiotic resistance in UK dairy cows.
Antimicrobial resistance, or AMR, is the natural resistance of bacteria to antimicrobial compounds, including antibiotics. AMR is a global challenge, with resistant infections causing 700,000 deaths. UK policy focuses on stewardship, using the right drug at the right time and the right dose, resulting in a 43% reduction in the volume of antimicrobials sold annually since 2015. The use of antimicrobials is essential in animal health, with the industry valued at $46 billion in 2019. AMR is a complex issue. My project seeks to distill the issue down to a local level and look at the picture in individual cows. Several factors increase the abundance of resistance, with antibiotic usage being the greatest. If we have a complete record of all antibiotics given to an individual, we can begin to understand what impact sustainable use has. This fits into the One Health picture of AMR, as resistance in cows can spread into the environment and impact our industries and health. 18 convenient samples were collected, with 508 colonies picked from TBX, classified as presumptive E. coli isolates. All cows that had received antibiotics received amoxicillin clavulanate acid, which is excreted in the urine. Susceptibility was measured by disk diffusion. When we look at the results of our susceptibility work, the results are good. Across 8 different antibiotics from 6 classes relevant to the dairy industry, overall the majority of isolates are sensitive and not multidrug resistant. Multidrug resistance, or MDR, is defined as resistance to 3 or more antibiotics, and we had 76 isolates out of our 508 present as MDR. I wanted to understand the susceptibility distribution of the isolates from individual cows to look for differences or similarities in the herd. Each cow is an individual, so it is difficult to draw strong conclusions from these results. On these graphs, each cross is the mean zone of inhibition for a presumptive isolate. The bar shows the mean of all isolates, and the error bar shows the standard error of that mean. The color of the bar displays the result, with green for sensitive, yellow for intermediate, and red for resistant. The graph on the left are the results from a cow that had received antibiotics 4 days before sampling. On the right, the cow did not receive any antibiotics 3 months prior to sampling. This provides some insight into the impact of antibiotic treatment. If we expand this to look at different lengths of time post-treatment, we have graphs here for 1 day, 4 days, and 14 days post-treatment. 1 day post-treatment, we can see there is ampicillin resistance and 2 intermediate results. 4 days post, we have 4 resistance results, and by 14 days, we have 1 resistance, 1 intermediate, and more spread of the isolates, suggesting that there is greater diversity in the susceptibility. While MDR is bad, transmissible MDR is worse. So how many of the MDR isolates can transfer their resistance phenotype horizontally? I carried out conjugation experiments with a lab strain with no mobile genetic elements and kanamycin resistance. A large number of my MDR isolates were streptomycin resistant, while my lab strain was not. So if I cultured them together, and there was a mobile genetic element present in my wild type, it would share that element with my lab strain. This would then allow my lab strain to grow on a plate containing streptomycin and kanamycin. Of my original 76 MDR isolates, 56 were resistant to streptomycin. Of those, 52 were sensitive to kanamycin and were used in the study, and of those, 10 produced successful conjugation. The question remained about the diversity and how similar these isolates were. I looked into ERIC PCR to provide a molecular fingerprint to these isolates and investigate diversity. Enterobacteriaceae repetitive intergenic consensus, or ERIC PCR, relies on ERIC sequences which are distributed randomly throughout the Enterobacteriaceae genome. This random distribution results in amplification of regions of DNA that vary in size. By electrophoresing the products, a fingerprint is produced. That's the theory anyway. I tried it and it doesn't work. This gel contains 4 different strains in technical replicates. The same DNA preparation was PCR'd, resulting in different banding patterns. Therefore, ERIC PCR cannot be relied upon to produce a molecular fingerprint. I would like to acknowledge everyone who has contributed to my work, those who have advised and supported me, and to the audience for listening to my talk.
Hi, my name is Priya Selvam, pursuing my final year PhD at Middlesex University. My research topic is an investigation on the effect of Salmonella typhimurium on root hair density in microgreens. Firstly, what are microgreens? Microgreens are also known as leafy greens or salad greens. It has a central stem, leaf or leaves, and roots. Typically, the first pair are of very young true leaves, whereas they are harvested within 10 to 15 days after germination. Initially, it was produced in the mid-1990s in the United States, beginning in Southern California, in a very limited amount, and then its popularity has been increased later. It is used worldwide extensively for garnishing in soup, salad, sandwiches, and plates. Salmonella is an important foodborne outbreak which is gram-negative facultative anaerobic rod-shaped bacteria that causes salmonellosis, and recently it is associated with the consumption of leafy greens because it can survive in the soil for a long time under all external factors such as low to high temperature, pH, and water activity. In this study, the effect of Salmonella typhimurium were observed in 3 types of microgreens, namely rhubarb chard, celery, and Swiss chard. And here my aim is to enumerate the number of root hairs of microgreen after exposure to Salmonella typhimurium and mutant strains. Here the graph shows all microgreens showed a significant increased number of root hairs when treated with wild-type Salmonella typhimurium. Y-axis is the number of root hairs per millimeter square, whereas the x-axis shows the number of root hairs per millimeter square in 3 different microgreens, namely Swiss chard, rhubarb chard, and celery. Here in each microgreen, the blue bar represents the root hairs measured in control plants, so they have not been treated with the bacteria, and the green bar represents the root or exposed to Salmonella typhimurium. Here to conclude that there is an increase in root hair density in all microgreens, but it much more pronounced in celery. The right side is the image of the same root. Here, the microgreens showed a decreased number of root hairs when treated with mutant strains compared to wild type. The graph shows here is the green bar is control and the orange bar is treated with Salmonella mutants. So the Salmonella is mutated in the genes that affected only the LPS here. To conclude, Swiss chard shows some significant results, whereas little bit less in rhubarb chard. But interestingly, there is no significant results in celery. Therefore, we conclude that celery responds strongly to wild-type Salmonella typhimurium. Hence, Salmonella root responds uniquely and distinctively. Interestingly, we found more root hairs in the treated compared to control. Mutant Salmonella do not increase root hair density in celery.
Hi, my name is Jordan Maven, and today I'm going to be giving you an overview of my research, which aims to improve the susceptibility of clinically relevant bacterial infections to antimicrobials. To do this, we use cold atmospheric plasma, which we shorten to CAP. Plasma is the fourth state of matter produced by the full or partial ionization of gases. So just to make sure that we're all on the same page, I created an animated infographic to introduce you to the main device that I work with, which is this one. This is our in-house developed kilohertz-driven jet. It can also be referred to as a dielectric barrier discharge system. As you can see here in the schematic on the right, the gas is insulated from the electrodes by the quartz tube which makes up the body of the jet. Helium and oxygen are flowed through to generate a plasma plume, as seen in purple, under which we treat our samples. The jet runs between 35 and 40 degrees Celsius, so it's not really cold, but we use the terminology to differentiate between these and thermal plasmas, which reach tens of thousands of degrees. The plasma core generates a diverse array of reactive oxygen and nitrogen species, as you can see in the diagram, which have a range of biological activities from stimulation of wound healing to antimicrobial activity. The unique combination of chemical and physical factors produced in the plasma effluent is key to its efficacy in eradicating microbial growth. The ability to treat biological samples with these cold plasma devices has ushered in a new era of plasma medicine, with recent work from our group demonstrating the potential of CAP for the efficient eradication of key pathogens with growing multi-drug resistance profiles such as Pseudomonas aeruginosa and the broader escape cohort. However, significant gaps exist in our understanding of the precise mechanisms of the antimicrobial action of CAP, which brings me to my work. During my PhD, I've been investigating CAP as a novel method of improving bacterial susceptibility to common antimicrobials in a world of ever-increasing antimicrobial resistance. While we see a subtle change using planktonic cells, the real benefit of the plasma is visible when cells are grown in a biofilm. To further understand the role played by extracellular components of the biofilm in mediating tolerance, I've been assessing the impact of sublethal CAP treatment on biofilms of Pseudomonas strains. I've included plasma survival curves here for 3 strains, I have Pseudomonas, an alternate overproducer, PA14, and a wind isolate, PA10548. As you can see here, plasma exposure causes up to 4 log reductions in cell viability after 5 minutes. As we're not aiming for eradication at this stage, the time point selected was 90 seconds, which affects a 1 log reduction. In combination with antimicrobial susceptibility assays, we were able to demonstrate improved sensitivity. I've included MBEC results here, as these are the most pertinent to the global issue of chronic wound infections. These are the concentrations needed for biofilm eradication in untreated and CAP pre-treated samples, and you can see up to a 256-fold reduction if you look at the concentration of gentamicin required for PaO1 here. Preliminary data using transcriptomic analysis and fluorescence microscopy suggest that the increased susceptibility is due to differences in the biofilm matrix. It's also interesting to note the differences in initial susceptibility. Future work seeks to further elucidate this improved sensitivity with regards to the differences between the strains tested. And finally, thank you for your attention. I hope I've provided you with an interesting overview of my research and the wider role of plasma medicine in society. Please get in contact with any questions or discussion. Thank you.
Dublin is host adapted, most often found in cows, but as a matter of fact it causes enteric disease in you and me and your mum. More likely to die with Dublin than with typhimurium. Infected cows and calves can spread the disease via meat and milk and poop and touch and their wheeze. The other reason that we look at Dublin with caution is it causes quite a lot of UK bovine abortions. Oh, salmonella is quite important, let me tell you. Could bring you to your knees, so stay away from unpasteurised cheese. Got 15 samples from the APHA, examined isolate growth so that one day we could use them to infect bovine repro cells called bovine cranicular epithelial cells, which really don't grow all that well. The cells are from the placentome, which you can see here in monochrome. Infected them to see how well Dublin survives in this particular cell line, and surprise, surprise, there was absolutely no statistical difference, so a year plus work without any significance. It's hard to be indifferent. Oh, Salmonella Dublin is genuinely interesting, and you can trust me on this, because I've been studying this pathogen for almost 4 whole years. Computational work has the benefit of showing that the isolates are representative of a wider population from various sources, so humans, food and dogs and sheep, as well as abortions. We looked at virulence and AMR and found something really cool and a bit bizarre. Human isolates lack a virulence gene, the first significant result of my PhD. Oh, salmonella is quite important, let me tell ya. Went to do my pip away, left the lab and finished writing to come back to it another day. Now the part where the song gets sad, just as I was due to get back in the lab to evaluate the host response when COVID hit, and pandemic PhDs are particularly shocking. So we look towards my future work. Provided my cells stop being jerks, I might finally see how BCECs respond to infection with this Dublin, and maybe beyond. We can use whole blood and monocytes to look at dissemination, and we just might be able to answer some of my original questions and submit my PhD with minimal corrections. Salmonella Dublin is absolutely fascinating, and you can trust me on this, because I've been haunted by this pathogen for almost 4 whole years. And some might say a song is not professional, but my argument to that is that a song is more accessible, and I know people switch off when I talk about cows, but you might remember stuff about Salmonella Dublin now. Hello Hello, I'm Gemma, and I'm a final year BBSRC DTP PhD student at the University of Nottingham, investigating why Salmonella enterica serovar Dublin causes abortion in cattle. Like the majority of scientists today, my work was significantly impacted by the COVID-19 pandemic, predominantly because of laboratories shutting down. Salmonella Dublin is a zoonotic pathogen, so I wasn't allowed to take it home and culture it in my Erin cupboard. So I spent 8 months of my third year out of the lab unable to generate data. Consequently, when it came to making my 3-minute thesis for an internal conference at Nottingham, I didn't feel as though I had enough data. My imposter syndrome got the better of me, and I decided it would be far less embarrassing to write and sing a song about my PhD than it would be presenting my actual data. People at the conference enjoyed the song, so I uploaded it to YouTube and shared it on social media. And one thing led to another, and now I'm here to talk to you about some of the work I've done as part of my PhD, and hopefully convince you that Salmonella Dublin is interesting. Salmonella enterica serovar Dublin is a host-adapted pathogen capable of causing a variety of disease outcomes in a variety of species. In humans, infection is thought to occur via consumption of contaminated beef and dairy products, and disease can be severe, often requiring hospitalisation. In cattle, it's the most commonly isolated Salmonella serovar in the UK. Infected animals may show clinical signs of infection or can act as asymptomatic carriers, excreting the bacteria in urine, milk and faeces, and contaminating the immediate environment with organisms that can survive for months at a time. Dublin is also one of the top 5 causes of infectious abortions in the UK, though its prevalence is highly likely to be underestimated. These are so-called spontaneous abortion events because they occur without the dam presenting any clinical signs preceding the abortion. Abortions incur economic losses via the necessity of veterinary treatment for the dam and her subsequent reduction in productivity, either in her ability to become pregnant again or a reduction in milk production. The dam may also need to be culled, potentially resulting in a loss of a genetically important animal, and of course, infection with Dublin poses a threat to animal welfare, and due to its impact on animal health, productivity and human health, is a food security issue. The initial infection occurs via the fecal-oral route, perhaps due to contaminated feedstuffs or even through cow-cow contact. The bacteria survive in the digestive environment and transverse the intestinal epithelial barrier to infect host macrophages. Salmonella, like a number of infectious bacteria, use a type 3 secretion system to enter and replicate within host cells. Type 3 secretion system virulence comprises a needle structure which protrudes through the bacterial inner and outer membrane and injects protein factors into the host cell which initiate actin reformation, forming the salmonella containing vacuole and engulfing the bacteria. The SCV can essentially hide the bacteria from the host immune system, and infection of macrophages likely facilitates the dissemination in blood to form infectious foci in a number of locations. Salmonella Dublin have been isolated from liver, heart, lungs, lymph nodes, and the reproductive tract, including in the placentomes of pregnant females. We don't currently understand what happens in this infection of the reproductive tract in order to cause the abortion. So part of my PhD has included investigating phenotypic virulence of 16 Salmonella Dublin isolates, 15 from abortions in the UK in 2017, and 1 reference strain from a case of calf diarrhoea in the bovine coruncular epithelial cell line, or BCECs. BCECs are a cell line isolated from the placentome of a pregnant cow. And the placentome is a button-like placental structure formed of interdigitating maternal and coruncular and fetal cotyledonary cells, which facilitate nutrient, gas and waste exchange during pregnancy. I wanted to know if there were any differences in bacterial invasion and survival in this host and tissue-specific cell line. So I cultured BCECs and infected them with multiplicities of infection of 1 and 10. I added 100 micrograms per ml of gentamicin 1 hour post-infection to kill extracellular bacteria, and 2 or 24 hours post-infection, I lysed the BCECs and collected the intracellular bacteria. After completing this experiment 3 times, we found that there were no significant differences between the bacteria in terms of their virulence or their ability to infect and survive in this cell line. This wasn't particularly surprising because in order to cause an abortion and therefore become a clinical isolate, the bacteria would likely have needed to infect these cells in the first place. I then wanted to know whether or not these 16 isolates used in the phenotypic studies were representative of a wider population of isolates, not just from cattle. So I sent my 16 isolates off for whole genome sequencing and collected 250 more Salmonella Dublin sequences from Interabase. These 250 sequences were all from the UK, isolated between 2001 and 2019, from humans, livestock, specifically cows and sheep, food, and miscellaneous sources, which included the environment and dogs. My secondary supervisor and bioinformatics whiz, Adam Blanchard, identified virulence factors using the virulence factor database and constructed phylogenetic trees based on core and accessory genomes. We've also done genome-wide association studies to identify single nucleotide polymorphisms of importance in livestock versus human isolates, as well as identifying antimicrobial resistance genes. The analysis of the GRAS is still underway, and there wasn't much time to talk about the AMR, but it was worth mentioning that we were doing both of these studies. The core and accessory phylogenetic alignments demonstrate that the 16 isolates used in the phenotypic study were representative of a wider population of UK isolates, and the tree based on the accessory genome shows distinct clustering of isolates from livestock sources. We identified 285 virulence genes, which were categorised into 23 functional groups, and from this we identified 46 virulence profiles. The functional groups included genes associated with type 3, 4 and 6 secretion systems, chemotaxis, ferric uptake, fimbriae, flagella and VI antigen production. Most excitingly, we identified virulence genes significantly associated with livestock isolates, confirmed using a Fisher's exact test. The presence of this gene is indicated by a red bar on the accessory alignment here. Interestingly, one of the isolates used in the phenotypic studies also happened to lack this particular virulence gene, indicating that this may not be absolutely necessary for infection and survival in BCECs. However, because of this strong correlation between the origin of isolation and the presence or absence of this gene, we're continuing to investigate its importance. Firstly, by completing whole blood survival studies using fresh bovine blood, which we hope will shed some light on its role in bacterial dissemination in the host. We're also going to conduct a similar virulence gene presence-absence study in Salmonella typhimurium isolates from the UK, again from a range of origins of isolation. And we hope that this study will reveal whether or not this virulence gene is specific to cattle infection and also whether or not it's specific to Salmonella Dublin. The remainder of my PhD will be finishing off the GWAS analysis and predominantly using the BCECs to investigate the host response to Salmonella Dublin infection. I'll be using quantitative PCR to quantify pro and anti-inflammatory cytokine production, ELISAs to look at prostaglandin synthesis, and also immunocytochemistry to assess cellular functionality and integrity during infection. With that, I'd like to thank SFAM for giving me the opportunity to present my work to you today, both in a song format and in a more traditional format. I'd like to thank my supervisors and laboratory group for their continuing support and patience. Thank you to the Animal and Plant Health Agency for sending me the 15 UK bovine abortion isolates, and to Professor Christian Farah for providing our lab with the BCECs. And finally, I'd like to thank the BBSRC who fund my PhD. And for anybody who might want to listen to the song again, just search Salmonella Dublin. It's interesting on YouTube. Thank you.